of discussions about the use of markers in the dissection of human traits and diseases. Has published a number of papers in linkage analysis, equilibrium analysis, association analysis, has a book out, the Handbook of Genetic Linkage, that discusses a suite of programs that one can use to conduct genetic analysis with molecular markers. So very fortunate to have Joe today. And so with that sort of introduction in mind,
okay. How do you turn this thing on? Okay. I just thought I'd start with sort of from the popular media. This is something from the Sun in England, which is sort of the general attitude I think people have about genetics and the power of these studies. And this is from the Sun, which is kind of a tabloid newspaper in England, but they say that some European scientists at the Lisbon Research Institute in Portugal have determined that chromosome 4 determines what work you'll do, chromosome 7 tells what kind of mate you'll choose, and chromosome 19 decides your financial future. With that in mind, I don't think we need to do any more work. And they did this all with 10 pairs of twins. And they found that the well-adjusted and happy twin followed their genetic game plan for fulfillment, and the depressed and sour one had strayed from what their genes had ordered for them. Now, this sounds a bit ridiculous to us, right? But if I showed this to my mother or someone, they'd probably believe it. And I think that this is really true, that the whole idea has gotten out of hand. I mean,
I hope you know that the Sun is the highest circulation daily.
Oh, I know, and I'm sure that probably a lot of people really do believe this, and I think that's also part of the reason why people are so alarmed about genetic information being used for this, that, or the other thing in their lives. I mean, I think most of us, the more we work with genetics, realizes that there's probably less and less that we know about our genes, and the less useful it becomes to think about it. And no, I don't mean it in that sense. I mean it's less relevant to be scared about these things and more to think about, well, the real problem is that we don't know that stuff about our genes. We don't really know what they do. Everything that we're talking about is all based on our best guesses and our best theoretical models. The same goes for how to detect these genes. I know President Clinton last week said that now you can map genes in 9 days. And that, frankly, was a bit alarming also, because I can't think of a single complex trait that's been worked on for more than 9 years that's actually, in case where these have actually led to the discoveries of genes, mostly simple ones that are rare and the standard methodologies work. So if this is the expectations that are really being broadcast out there, we have to be a little bit careful. Now, with that in mind, we all know this part here, what a gene looks like. This is just some basic slides at the beginning, and then I'm going to go through the details on the board. Okay? Oh. Okay. Anyway, this is what a gene looks like, and you can see there's many different places within a gene where you can have mutations that can have different effects on any phenotype that you're interested in. I mean, most of the things that we're approaching in our studies tend to concentrate on these regions here where the actual coding sequence is, but there's good reason to believe that there may also be mutations or variations within these genes that can affect your phenotype that are in these various regulatory regions and other regions that aren't even well identified here, because we don't really know where these things are or what the genes do. In a lot of studies in Drosophila, for example, when there were people looking at Drosophila bristle numbers, they found a large percentage of mutations to be in upstream regulatory regions rather than in coding sequence. So it's just something to think about, that just because we say we have a mutation in a gene that causes a disease, we don't mean that the mutation has to occur in coding sequence. It could be in an intron, it could be upstream, downstream, anywhere. We just don't know. And that's why we're trying to find out. I mean, initially when we were looking at monogenic diseases, the idea was that we would do linkage analysis, we'd collect a bunch of markers, look at it, and then we'd find out what the gene did or what it looked like where it was. And that would also ultimately prove that it was genetic, because a lot of times things look genetic and they aren't necessarily so. I guess Kuru is a good example where it came from eating your parents' brains when they died or something. And that's how it was transmitted, and that's familial. It's transmitted from parent to child. So things can misleadingly look genetic when they really aren't. So this is the idea. A linkage analysis, a positive result, proves there's a gene and shows you where it is. With complex traits, it's a little more difficult than that, yeah? For example, just to think about the complexity of disease, this is from a book by Ken Weiss looking at the lipid physiology system, and you can see that there's numerous different, this is a very simplistic model of the system, and yet there's numerous different pathways where genes could be messed up, destroyed somehow, and give you a phenotype that affected this more general trait, yeah? In fact, if you look at some studies looking at systolic blood pressure, for example, you can see that you may have estimates of heritability that are fairly consistent across all kinds of different populations, you know, from some island residents to Japanese Americans, French Canadians, Brazilians. And when you see these things, my first reaction is a little bit of concern, because while you are seeing consistently high heritability, the fact is these populations are not necessarily very genetically related. So if the heritability and or the prevalence of these different traits in different measures stays constant when you're looking in all different regions of the world, you can imagine that we're looking at something where if there are very important genetic components, they're probably very sort of common components that don't have big effects, or there's many different places in the system where things are broken up in different regions and some sort of evolutionary pressure has maintained the level of this phenotype in a range where it can be tolerated. And now, when we think about these genes also, it's very important to consider how often we find mutations within genes. Now this is, again, this is from that same book, just because it was handy when I was looking for something to photocopy down to overheads for this purpose. This is a look at type 1 collagen genes and various mutations where they were located and found in the trait. And this is where, in the physiology, where these well-known mutations affect the receptor function. So you can see, in this one gene here, you can see there are several different mutations listed out here. Some of them are lethal, some are severe, some are mild, but they all affect the general phenotypes in different ways, right? So the idea that I want to get across here is that when we're looking for genes for more complex diseases, and I would assume, since this is a Center for Environmental Genetics, where most people are not so interested in finding genes that cause something that's rare, very strong, very obvious, and doesn't have environmental components, yeah? Looking for things that are more common, more general variation. And when you get into these genes, you're going to deal with a range of mutations, probably, in many cases, that have a range of effects. It's not necessarily so simple as we think it is. I'm just putting all this stuff up now at the beginning so that when I get into the talk, you'll see sort of why I'm saying some of the things I'm saying. And I'm going to present the methods for detection in a way that's kind of independent of what the cause is and everything else. But I want you to think about these things while we're going through the methods for detection. To think about, you know, what's the ramifications of this on a different approach. Again, this is looking at, again, mutations in the LDL receptor locus. And again, they're in various different places here. These are deletions and point mutations. There's quite a few of them characterized. I think most genes that have been studied in detail have shown huge amounts of variation, huge different numbers of mutations. The other big problem that we have when it comes to genetic studies is a lot of these studies are being conducted by people who may or may not have had very much training in genetics. And as you can say, you know, after 5 years of college to be a CPA, 6 years to be a technical engineer, 6 months on the OJ jury to be a DNA lab technician. And this was obviously a joke being made at that time about how much technical detail they were going into, but I think it says something else. I mean, oh! Sorry about that. The point I think that's important about this is that there are a lot of people starting genetic studies. I know, being at my institution, I have people coming all the time. They have various clinical populations. They see some people where they have a couple SIDs that have this disease, and they immediately jump and say, ah, genetics. President Clinton says we can map genes in 9 days. Let's go and try to do this, because it's sort of the hot thing right now to try to do genetic studies. But there's a lot of people who are trying to do them who haven't taken the time to understand the basics. And I think that these statistical basics are probably, in many ways, the more important ones to have a grasp of, because that sort of tells you what can work, what can't work, why does it work, and what are we doing, yeah? Okay, and this is another table from another population genetics book looking at approximately minimum numbers of genes involved in some quantitative traits from lower organisms where they can actually do studies to figure this stuff out. In humans, it's rather more complicated. it's not so ethical to say, okay, you and you, I want 10 kids and I want them next week so I can study, their offspring and their phenotypes. We have to look at, examples of matings that have happened naturally, and we have to sort of try to evaluate observational, data instead of experimental data. But here, you can see that in these cases where they know it for these various traits, there's a large number of genes involved. Now, hopefully, we're going to be finding things that are a little more unusual, a little simpler, and we'll be able to identify them more easily. Okay, and this was based on similar factors. When we have these quantitative traits, there's probably a distribution of effect size of these different alleles in the same gene on a phenotype. Okay, so this is from some experimental data showing somehow strength of effect of a given allele on a phenotype as a function of what allele it was and how frequent that allele was in the population. The punchline here is that obviously, if you had a very common allele that had a very huge effect, everybody would be sick if it was working at a disease phenotype. So if you have something that's fairly common, has fairly low recurrence risks to siblings or to first-degree relatives in the family, we're probably thinking about genes that if they're common, the effect is small. If they're rare, the effect is large. But if they're rare and the effect is large, it's not very useful for most people who don't have that very rare allele because it doesn't have that much to do with their disease, although it may give us clues into the general way that the disease operates, that sort of system operates. But we're basically trying to find things that are fairly common and don't probably therefore have very strong effects. Not good news necessarily, but it's good news for those of us who have those bad alleles because we're not that sick. And I already showed that. That's right. Okay, now I want to use the blackboard and discuss what basically it is that we're trying to do. Okay? The reason I'm going to use the blackboard is because when I use overheads, I tend to talk fast and I don't explain things thoroughly enough. And I think that's pretty common among people that tend to do statistics and math. We sort of skip steps in our head when we see the results on paper. When we have to write them out, and we have to think more, and we have to make it more understandable. So that's the reason I'm doing this. Now, basically, in all of these studies, we have different effects. We have our gene. If we're looking in any kind of a genome scan or any other procedure, we're only looking for one gene basically at a time, because one gene's at one place, that's the one that we're trying to find the effects of. So that's the gene of interest, has an effect on the phenotype. Now, there's other genes too, probably which affect it, and then there's environment, obviously, and various other factors. You can have various interactions here and here, whatever. Okay? Problem is, this is not what we have to study. Okay? We don't have the gene usually. And even if we have the gene, we probably don't have the right allele. And even if we have one right allele, there's probably a lot of other alleles that are also doing something. So we never really have this. And even if we do, we don't know we have it, so we assume that we don't. What we have, well, that's real. I mean, it's very hard to actually prove that you found a mutation that's caused it, because it's probably, as Nick was saying this morning, it's probably associated with something else that's very close to that point. Another marker, if you will, another one of these loci that's right next to that gene that could also have exactly the same significance in any kind of statistical test. You have to use more basic biology to prove you've got the gene. You can't really do it with math. We can just give you, evidence or, probabilities, but that's never probability of one. And so what we have are these guys, these markers. Right? These markers are correlated with the gene, but they're not the gene. They're correlated with it somehow. Okay? And we're looking for this effect here. That's the only thing that we can detect, at least it's the only thing we're interested in detecting in a genetic study. Okay? In an environmental study, you want to emphasize this factor, but in a genetic study, you want to emphasize this. Now, we can do various procedures and various study designs to figure out ways of maximizing these correlations. You can either attack it here, or you can attack it here, because you need those two to be strong. On the other hand, you want those effects to be weak, so that you can emphasize the correlation between the markers and the phenotype in your sample. Okay? Because that's our goal now. Our goal is to find one gene that's doing something per se, okay, at a specific point. Maybe we ultimately want to find these guys too, and we probably do, and we don't know which one we're looking at. But the point is that we're looking to find evidence of a gene correlated with a set of markers, okay? That's really the fundamental goal. So, how are markers and genes correlated with each other? Well, within pedigrees, as you've seen, there's the phenomenon of linkage. If the marker is here, this is a chromosome. I tend to think of a chromosome as a big long line and genes as points because that's the simplest mathematical representation. I can't really draw what they really look like. Otherwise I wouldn't be a mathematical person. Okay, so here's the disease locus, here's the marker locus. There's some distance between them, the recombination fraction, which we denote with the probability theta. Okay? This is the probability of recombination occurring between a given marker and a given disease locus. Fair enough? So if they're at exactly the same point, this value would be 0. If they're on different chromosomes or unlinked, then that would be 50%. Because you have 2 chromosomes in your parents, and you get 1 at random, and there's 50% chance of getting either one, independent of the disease. Okay? So this range is between 0 and 0.5. Now, that is in one way that markers and genes are correlated. The other way that's been explained this morning, the other type of a correlation between markers and a given gene is through linkage disequilibrium. Okay? You've been introduced this morning to how a founder effect can give you some linkage disequilibrium. I'll go into more detail about the dynamics of these two processes in a little bit. And that becomes more important when we discuss the main goal, and that is how do we increase these effects? Yeah? We want to sample in a way where we can maximize these correlations. Okay? But these are the 2 ways that they're correlated. So they're not independent ways. Linkage disequilibrium, as you've seen this morning, is correlated strongly with the distance theta. The closer a marker and a disease are together, the more likely that they've been inherited. Given a allele in a marker locus and a disease locus, are more likely to be inherited in common over many generations when there's a very small chance of a recombination breaking up the disequilibrium. Okay? But those are really the 2 main factors. Now, they do occur at the same time. If there is linkage disequilibrium, it will affect your pedigree analysis as well, because they'll be more likely that in different pedigrees, it's the same allele that's co-segregating with the disease. Okay? But in any event, those are the 2 main effects. Now, we also have the correlation between the gene and the phenotype that we can do something. Well, we can't do something about it, but in sampling we can. Now, how can we do anything about strengthening this pathway? Well, you can obviously find various subphenotypes that are more unusual, more heritable, if you will, in which the effect of genes is stronger. So everything that we do in terms of trying to design a good study to find these genes causing that have some effect on some phenotype is based on those increasing. Meanwhile, decreasing the effects and gene by environment correlations wherever that's possible.
That's good.
Yeah.
Question, just terminology. Disequilibrium, is that identical to linkage? Is that a buzzword or is that a real word?
Okay. Basically what it means is this. Let's assume we have 2 loci. Okay? We have a marker locus and a disease locus. At the marker locus, we have 2 alleles, 1 and 2, for arbitrarily, okay? And at the disease locus, we'll have, let's call them 2. Let's say that there's 1 disease allele D and a wild type allele plus. Very simple model. What it's saying is that the alleles that occur at the marker locus are independent of what allele occurs here, such that if you did a 2x2 table of them, these occur on haplotypes, if you will. In other words, you have a marker locus allele and a disease locus allele that you can observe jointly. It would be saying that if you have at the disease locus the 1 allele at the marker locus and the disease allele at the disease locus, this haplotype, it's basically saying that it's independent, that these are independent.
So complete disequilibrium is no recombination?
Disequilibrium has nothing to do with recombination exactly. It's a function of the prior probability of this haplotype in the population. In other words, if I just picked you out of nowhere and I didn't know your parents or anything about their genetics, it's the probability, given that you have the disease allele, the probability that you also have the 1 allele would be the same whether you have the disease allele or the plus allele. It's a correlation that, as Nick mentioned this morning, if you assume that initially in a population you have haplotypes 1 plus and 2 plus only, okay? There is no disease yet. Now, a disease allele enters the population through some mechanism, and let's say that it happened to occur on a chromosome with allele 1. Then there's going to be too many of these haplotypes initially. There'll be none of these at this point in time because all disease alleles occur on the chromosome with allele 1, okay? Now, over time, you will have, if these are separated by distance theta, there's a certain probability that you will eventually have a recombination in an individual who has this genotype, okay? Now, a recombination in that individual between those two loci would generate a haplotype 2D, okay? Linkage disequilibrium merely means that there is some difference in the distribution of allele frequencies on diseased chromosomes and plus chromosomes. This is the most common model assumed for how it's generated, okay? Linkage equilibrium means that there is not such a correlation, okay? It has nothing to do, per se, with linkage. Well, I mean, it does in the sense that if they're tightly linked, you'll have this equilibrium more often than if they're not. Because if they're not, this will be broken up very quickly, okay? But linkage, rather, means this, that within a pedigree, let's say, for example, that we knew this person had genotype D1 plus 2. Okay? And let's say we happen to know that the disease and the one allele were on the same chromosome. Linkage means merely that this child, who has also received the D allele, is more likely also to receive the 1 allele because there's a small recombination fraction. That's looking at it in the sense where you're actually observing meioses. In the linkage disequilibrium study, we do not observe meioses. We observe the outcomes of many meioses many generations later. So we're only seeing the haplotypes at best. Okay? Is it clear basically what those two phenomena are? Now remember that when we're looking at any of these, this disequilibrium and linkage are a function of the loci, the alleles at the loci and the correlations between the alleles at the loci, either in the population sense or in a pedigree sense where you can actually observe the combinations. Okay? It's not phenotype. Phenotype is nowhere involved in any of this stuff yet, okay? Phenotype, right now what we're looking at is this stuff, markers and gene correlation. The phenotype is here. This is a completely separate correlation, okay? It's what is the gene's effect on what you can observe. In other words, does this gene increase your risk of, cancer, heart disease, whatever? That's something independent of linkage and independent of association. Okay, that's a function of alleles at loci. We're trying to use that where we only have this as a proxy for that. It's kind of sort of like if you think about the Framingham heart study where they were looking at coffee drinking, smoking, and heart disease. If you think of coffee drinking, the gene says smoking, and the phenotype is heart disease. Most of the correlation with coffee drinking and heart disease was because coffee drinkers had a correlation with smoking, right? And it's a very similar sort of situation. We're looking at a proxy for the real risk factor. We don't have the real risk factor because we can't have it. It's just something correlated. Kind of like saying, if this is breast cancer and this is being a woman and this was some other phenomena like didn't play football in high school. Therefore, people who don't play football in high school are more likely to get, or who did not play football are more likely to get breast cancer. Yeah? But that's all the data we have. We don't have that they were women. We weren't allowed to ask that because NIH wouldn't let us. Didn't get into our affirmative action. But really, this is always the case, and it's very important to think of that because this genotype to phenotype correlation is nothing different from any epidemiological study. This is a risk factor. This is something that it's creating a risk for. All the problems come in here. These correlations are caused because we're looking at, various observations and pedigrees that are not independent individuals. They're all related, okay? And because we do not actually have the real risk factor to look at, we only have a proxy for it. Now, the important question is, what do we do here and how do we increase the power of any of these studies? And I want to go through this a little bit, as I said before, I talk about the statistical details of the methods, because the methods themselves, I mean, there's programs that'll do the number crunching for you. What's important is to understand the properties and why and how you can do something about this when conducting a study. Because I tend to think that the biggest place where we as statistical geneticists can input an effective project is at the onset, when you're trying to set it up and design it in such a way that you have a realistic chance to find something. Yeah. Now, when we're looking at correlations between markers and disease, how can we increase and maximize those correlations? Well, as Nick mentioned this morning, if we're looking at linkage or linkage disequilibrium, whatever, if you have more markers, you have more chances that one of them is actually correlated with the disease, assuming that there is a gene in these families that we can detect. Yeah? If I only looked at 10 markers in the whole genome, there's a very good chance I'm not going to find something, right? But if I looked at 1,000,000 markers, if there's something to be found, my chances are much higher. Of course, at the same time, there's an increase in the false positive rate if you do too many tests. So there's some trade-off. And I would bet that ultimately, if we were to take the total number of markers and the power, this is number of markers, there's probably some maximum after which it falls off a bit, because when you get too many tests, you're eventually going to find things that aren't really there, because the number of possible tests is greater than the number of possible data points if we had the whole world as a sample. So it's something to be a little careful of. But there is some happy, important point to that. The more markers you have, ultimately you have a chance to find something if it's there. Okay? Now, how else can we increase the power of a study to find linkage? Well, what is linkage? How does it work, these tests? Well, what we have normally, if we take the simplest case of a pedigree, let's say that we had a sibling pair. That's two siblings who are both affected with a disease, right? Now, if there's no correlation between a genotype and a phenotype, those two guys are going to share half their alleles in common, yeah? So let's assume for the moment that we knew the parental genotypes at the marker locus to be 1, 2, 3, and 4, and we knew the first child was a 1, 2. Just for arbitrariness sake. There's no evidence, no information here tells me anything about linkage yet, right? Because I've only put one thing down and there's no way to tell if this what's a recombinant, what's not a recombinant. Just one piece of data. Now, this child could be 1, 3, 1, 4, 2, 3, or 2, 4, right? And each one of those is equally likely if there's no correlation between being affected and the marker locus. Okay? So again, what we're looking at so far here is this phenotypes and markers, right? But we're only finding something because there may be a gene there. If there is a gene there and they are linked, then they're more likely to share alleles, right? That's the whole basis of linkage studies, is that we can find things where affected individuals have more genetic similarity than random individuals. If we didn't know their phenotypes, everything would be equally likely even if there was a correlation. Yeah? Okay. So, now, that doesn't seem like there's very much there, though, because we only have, from this one family, we only have really one observation. Because this one's genotype doesn't really tell us anything, because we have nothing to compare that person to in a linkage study. Okay? Remember, what we're trying to find is that affected individuals share more than we would expect them to share if there was no linkage. This, if we just have genotypes and one person being affected, there's nothing to compare that to, yeah? In a linkage disequilibrium study, however, there would be. So let's look here at the situation. I should have started from this situation, actually. If we have one individual down here who's affected, it can have genotype 1, 3, 1, 4, 2, 3, and 2, 4. Each one of them being equally likely. Linkage isn't going to affect that, is it? Is there any way that a correlation between this marker and a disease caused by linkage alone can affect it? Well, let's assume the most simple situation where these parents each were informative for the disease locus. Okay? They're both informative. Now, let's say we even knew the phase, and we knew it to be like that. So this person came out 1, 3. Now there's information. Isn't there? If this child is 1, 3, and we knew the phase where the parent was 1 plus here, and this parent was 3 plus. Well, the probability that this child was 1, 3 would be 1 minus the recombination fraction here, because this one has to be, if this one was DD, this is a rare recessive disease, the simplest case scenario. Rare recessive, parents unaffected, child affected. If we knew the phase, this would just be 1 minus theta times 1 minus theta. Okay? Give that square root, square root. Now that's information, because if there's no linkage, the probability of this observation would be 1 minus a half times 1 minus a half equals 1/4. But if theta here is 0, that probability is 100%. Okay? Is that reasonably clear what I'm talking about there? Now we normally test these kind of measures using some sort of a likelihood ratio statistic. We basically compute the probabilities of these observations up to some constant. So we compute the likelihood as a function of theta and divide it by the likelihood when the recombination fraction is a half. So in this case, the likelihood of theta is going to be 1 minus theta squared, probability of the linkage information, times sometimes some constant here for the frequencies. And in the denominator, it's going to be the same constant times 0.5 squared. So there is information there about theta, right? We all see that? Now let's say I didn't know, as I wouldn't know in a linkage study, what the phase is here. Okay? Now what do we have? One individual looking at linkage alone. There's now 2 possibilities. This person could have either 1 plus 2D, or it could have 1D, 2 plus. We have this first combination, and there's the same for this individual. Why don't I just make this individual uninformative for now? Just so I don't have to do the computation twice. This individual now has no information about linkage because this person's homozygous at this locus, and every child of his has to receive the allele number 3 with 100% probability. So this theta term is now gone. This 1 minus theta term, as is that one. However, when we have these 2 different options here, they're each equally likely a priori because we don't know anything about the correlations between the genotype and the phenotype. I mean, between the genotype at this marker locus and the disease locus. There's no correlation between them. There's no linkage disequilibrium here. What we have then is 1/2 is the probability of this phase times what would be then 1 and D being a recombinant in theta, plus 1/2 being the probability of this genotype for that person, times 1 minus theta. Well, that just equals a half, and there's no theta term in there. So there's no information about linkage from a single individual unless we know the phase. And when we add the second individual here, there's the correlation between these 2 that will give you information. Okay? If there's a correlation between genotype and phenotype, and the gene and marker are correlated here, we expect these guys to share more than half of their alleles. Okay? Now, the more SIBs you have in a SIB shift, the more information you have. Because if we have another SIB here, and this is a recessive disease, and that one's also 1/3, the probability of observing that by chance is very small. Here, note that what we have is a situation where this parent is 1 plus, and we don't even need to look at this now for the moment. This is 1, 2, okay? The probability of this one being 1, getting allele 1, is a half, right? If there's no linkage. The probability of this one also getting it is also a half, if there's no linkage. The probability this one's also gotten it is another half. So you multiply this together, and you get 1 in 8. So 1 in 8 is the chance of observing that pedigree when there's no linkage, right? Now, if there is linkage, and the recombination fraction is 0, what's the chance of this? Well, again, we have 1D, 2 plus as a possibility here, or we could have 1 plus 2D. We're thinking about this in the simplest terms of a recessive disease where we know this. Now, if this is the case where we have 1 and D together, we're going to have a half is the probability of that, a half is the probability of this being the phase here. And then it's 100% chance that each one of these got 1 in D, because there's no recombinations allowed, right? If it's complete linkage. So this is just going to be a half times 1 plus a half is the probability of this phase. But if you have this phase, it's impossible to get a 1 in D on the same chromosome, yeah? You can't have recombination if the recombination fraction is 0. So that's going to be a half times 0. So the probability of this outcome, given this information and 3 affected kids, is going to be a half when there is linkage. So our likelihood ratio is going to be a half divided by an 8, which is 4. Okay? So there's some information here about linkage, and there is some power to detect it. As we add more sibs here, if I add another sibling out here, I'm basically not going to change this probability, right? This is, again, remember, 1 minus theta to the nth power, where n is the number of sibs, each sib being independent, okay? And the denominator here is 1/2 to the nth power. So our likelihood ratio is going to be 1/2, 1 minus theta to the n from this function divided by 1/2 to the n. Okay? So as we add more sibs, that number is going to increase and increase and increase, and we're going to have the ability to detect the linkage. Now, the point here that I'm trying to make, which has been rather convoluted by all of this algebra, which isn't exactly the most interesting part of it, is that when you have a simple disease that's relatively rare and has a strong effect, you want to get your pedigrees to be as large as possible so you have as much information as possible. Because we aren't assuming that we have information about linkage disequilibrium, only about linkage. Linkage being a measure where you get one observation of each meiosis, and there's no information when you have only a sib pair. Remember, you're only going to have really one observation that helps you detect linkage, because the phase is unknown for this case, right? Yeah.
You've described this only using the children with disease. Is that,
Yeah.
This analysis is restricted?
It's not, except for this. So far, I'm trying to look at ways that you can approach a correlation between something that's rare, the disease being a rare phenotype, I'm assuming in this case, and when you have if you have unaffected siblings of these guys, right? This unaffected sibling, if it's a rare recessive disease, could be either plus or plus P. And there's not that much information to discriminate between the two. So these don't provide very much information about the linkage study. When we get to more and more complex phenotypes, the correlation's even lower. Just that affected individuals, you may want to assume that if you're affected, there's a greater chance that you're carrying some disease predisposing allele at some locus. But you may not be as willing to say that an unaffected sibling doesn't carry it, unless there's something unusual about this unaffected sibling. For example, if these guys had extremely high cholesterol, if this unaffected sib had like extremely low cholesterol, that's a very different phenotype that's also rare, okay? The point is that you need something unusual, something rare, in order to detect this stuff easily in these kind of simple approaches, okay? And that's what I've been focusing on so far. For a simple, regular disease, you increase power by increasing size of pedigrees, by increasing the affected information that you have when it's unusual to be affected for other reasons. Okay?
If it's really rare and simple case that having 4 of the same kind is also rarer than rare.
No, because they have correlated genotypes if they're in families. So, for example, in Finland, they've studied a lot of these recessive diseases that are very rare. They may have a frequency of 1 in 10,000 or something. But you see them all in the same family because they're caused by some allele that's very, very rare. But once this person has that allele and this person has that allele, 1/4 of their kids are going to get both of them and be sick. Okay?
They're not independent.
They're not
Selection
independent.
out of the sample.
Yeah. Disequilibrium, on the other hand, you're looking at population data where it is unusual to be affected, perhaps, and it's harder to ascertain a sample. But when you have relatives and the phenotype is common in the pedigree, rare elsewhere, that's when gene mapping has worked so far very powerfully. Okay? But as you say, you're right. You don't want a situation where these four have a good chance of all being affected for reasons that have nothing to do with genetics. If that's the situation, you're in big trouble. Because, as I said, we're interested in having this correlation be very strong, and so far I've assumed it is. Okay? This we can do something about easily by having more markers. We can decrease data, this recombination factor, as much as we want. But we can't do anything about this very easily unless we find some phenotype that's rare, common in pedigrees, rare in the population. Yeah? That's sort of the way it works. Because genes, I mean, we're looking here so far at rare genes with strong effect causing this phenomenon. If, on the other hand, you have the phenomenon where the genes are common and phenotype is, common, it's very difficult because you're not going to have such an extreme correlation here. You may very well have 1, 3, 1, 3, 1, we don't have that in this case. There's a 2, 3, and maybe this one might also be 1, 3. Here you may have 3 out of 4 sharing an allele, but one that didn't, or something on that order. You're not going to have a phenotype, genotype causes phenotype situation. Okay? That's really the difficult part about modern genetics, is that we don't really know what the correlation is between a genotype and a phenotype until we find the genes. But we can't find the genes if we don't know there's a correlation between phenotype and genotype. So, what do you do? Good question. But anyway, the point here I'm trying to make is not focusing on this, but focusing rather on this. Okay? We want to increase the amount of information we can get in our linkage study, therefore increase the correlation between markers and the gene. You do that by increasing the number of markers, and in the case where the gene is rare, by increasing the number of affecteds in the pedigree if there aren't a lot of environmental causes. Yeah? Okay? That's really the point I was trying to make about this. Now, it's also useful to have pedigrees with multiple generations. For example, let's say that this person's unaffected, but happened to have an affected child. If we know it's a rare recessive disease, then we also know, well, that can't be plus genotype. It has to be plus D, because in order to have an affected child with genotype ED being rare recessive, this one therefore had to carry the allele. So when you have larger pedigrees with many generations, there's a lot more information you can get out of them when the disease is rare and it's caused by a single gene. Okay? So, and this has been exploited. I mean, if you look at these genes for breast cancer, which nobody would have initially probably assumed to be having rare genes with strong effect, they did happen to find pedigrees that were multiple generation pedigrees in which these alleles happen to co-segregate with the phenotype, and that's how the BRCA1 and I guess BRCA2 genes were mapped. It was using multi-generational pedigrees because it's unusual to find multi-generational pedigrees. That was a way, perhaps, of increasing the genotype-phenotype correlation by finding some unusual characteristic of the disease in a subset of data. This has worked well. This whole approach has worked very well for mapping a lot of genes of rare, that are relatively rare and have relatively large effects. And it's been done, in fact, this is probably the only, so far to date, genome scanning approach that has actually led to cloning genes that I know. Do you know of any others, Nick? Nick knows the literature a lot better than I do, so if there is something, I think he would know it. But it really hasn't, in these cases it has. So it's useful if you find pedigrees that have multiple generations. There's a good chance that there's something unusual about that phenotype. It may not be the phenotype that you were looking at in general, or I mean, it may not have exactly the same genotype-phenotype correlation that you have in the real general population, but it might be interesting to look at those, where there's a good, not good environmental reasons to consider them. That's another factor, I think, that's also been something I've been concerned about. In a lot of these studies, it's very possible that you happen to have cultural or environmental factors that can be transmitted in pedigrees, much like genes are, like in the case of Kuru, where you eat your father's brain when he dies. That'll give you something that looks genetic. But likewise, there are other factors, like, say, smoking, which seems to be correlated among relatives. And I mean, if your parents have, stuff around the house, you probably have a greater chance of using it than if they didn't. I don't know if that's the case or not. That's just an arbitrary thing I'm pulling out of my head. But there are factors that have relatively large effects on diseases that are inherited, that are dichotomous in the population, that can look like genes in segregation analysis. So there may be other reasons that you observe a large multi-generational pedigree, but those would be something that would catch my eye if I was interested in doing a study, if I happened to run into them. For that reason, there's a lot of information there when that is the case. As for other ways to increase correlations between the markers and the genes relative to a linkage study, well, it's not such a simple thing to think of. And I think a lot of studies have concentrated on only collecting SIV pairs, for example, just these guys. With the argument that it's much easier, one thing is it's much easier to apply sophisticated statistical methods to this simple data structure because the correlations are much simpler to quantify, write out, and everything that it is to do something in a general pedigree situation like this. You can't apply real sophisticated, complex types of analysis to very complex types of data and have any real understanding of what's happening, at least not in a lifetime on current computer systems that I know. You can do approximate methods that work fairly well on these type of pedigrees for truly complex phenotypes, but you often can't for others unless you have some simplifying model, like I'm only going to click this kind of data or something like that and just get thousands and thousands of these guys, and hopefully the sample size issue, increase in sample size, can take care of the loss of power due to the fact that you don't have so much phase information and you don't have so much other reasons to think that there's something unusual about the phenotype in that family, because you have a large sample of these. Another reason why people might collect these instead of these is because it's a lot easier. I mean, it's easier to find two people who are siblings that have some phenotype than it is to find 10 or 20 in a large pedigree. That's sort of your argument that you made before, that it's easier, it's much more difficult to find these guys. But if it's that much more difficult to find these guys than these guys, then that probably means that you're not looking at anything that has a really huge genetic effect, and you're not looking at anything that's particularly rare. So you're going to need a lot of these guys, if that's all you can find. My basic approach to telling people when they come to me what kind of pedigree should I collect, et cetera, and I just always say, well, collect what you can find. Because if the gene is rare and has strong effect, you'll see these guys. And if you see these guys, you don't want these guys, because if you see a lot of these guys, it means there's something different, something going on that should indicate that's what you want to study. I've seen a lot of, seriously, there's been a lot of debate about these kind of points, I know, in the literature, and I think part of the problem is if the question is asked, what kind of pedigrees are most powerful for linkage analysis? Well, if you do the simulation study where you don't simulate the pedigree ascertainment as well, you're going to come up with answers that probably indicate something different than what I'm saying. I'm just thinking that if the phenotype, the way you simulate the phenotype leads to this kind of pedigree occurring very frequently in the real population, in other words, if you simulate population data and ascertain it, probably you'll see the answer to the question more simply than if you said, okay, would I rather have 10,000 of these or 500 of these? Yeah? It's a very complicated thing to think about what's really going on. Since none of us know what the true model is, we can't even simulate this question, but we can guess. Yeah? And I think that in most guesses, the right answer is going to turn out to be whatever is actually in the population is the best thing to sample, because it's also the only thing you can sample. Okay. Anyway, enough about this aspect of things. The other question that's important is, relative to this, has to do with linkage disequilibrium. How can I increase the chances of finding linkage disequilibrium and having it actually exist between markers and genotypes, the markers and disease genes in my population? Well, one thing is you can obviously look for populations that have less variability. Small populations where it's very likely that all the different people today who have a certain phenotype have the same certain gene, certain mutation in an allele, which came from a common ancestor not too long ago. So if you have something like in some of these real small island populations, let's say we were going to Pitcairn Island or something where they had 7 or 8 founders or something, and there's a disease there that's fairly common, well, then they're probably, everybody who has it today can trace themselves back to that founder person, these guys being today's effectives. If they can all be traced back to a common founder not too long ago, that at this given disease locus, in other words, this is a mutation here, and they've all gotten it from this guy back here, they'll also have a certain region around that gene that's also been inherited together with it, as Nick explained with much better drawings this morning. if you can find this kind of population, that's probably better than going out to Cincinnati, standing on a street corner, and taking the first 10,000 people who walk by, sampling them, and seeing who's fat, or the first 100 real fat people and the first 100 real skinny people, and saying, okay, I'm going to take these guys and genotype them, and that's going to be my association study. If you have this weird island population, and they also happen to have a large number of people in both ends of the distribution for the phenotype you're looking at, you're going to have a much higher chance of finding linkage disequilibrium because they're more related. In fact, in those contexts, linkage disequilibrium and linkage become the same thing, right? Remember, here we're looking at correlations between genotype and phenotype caused by the fact that in this case 1 and D occurred together. Okay? This 1 and D going to here, the reason they stay correlated is because they're linked. There's not much distance between the two loci, so the alleles of the two loci are co-inherited. When you go to this kind of population, you're doing the same thing. It's as if you had like a pedigree structure here, but at which none of the individuals were genotyped. Okay? So in those contexts, it's the same. If you happen to do that using everybody you run into on the street corner in Cincinnati, you're probably not going to have this structure where the individuals can trace their disease mutation to a common ancestor. That's not so likely. Remember that, as I showed you a little while ago, most of these loci that have been studied have, but there's tons of different mutations in the same genes that have very similar effects in many cases. So it seems unlikely that the same exact ancestral mutation in the same exact gene is going to be causing, me to have my eye color and, someone else who has the same eye color to have theirs if they're not from a similar ethnic background. Now, there are cases where you have alleles that are very common, that are maintained, that are very ancient alleles that came back even, might even be shared from other earlier evolutionarily related organisms. You may have, in fact, the same allele occurring in mice or something as humans. It's not necessarily, when you see that, that's probably the same mutation. It's probably the same event that you're observing in different individuals in the population. But the distance back to this point is so large that you may not be able to see any residual correlation remaining in terms of the markers that are in nearby alleles, okay? It may be possible if you get close enough that there may not have been recombination, but that's not very likely in many cases, that there's been no recombination and no anything in these genes. This other guy, Ken Weiss, is a researcher. He came to Columbia a few weeks ago and gave us a talk where he actually looked at inside a gene. He had a 9 kb region, partially intronic, partially exonic. And this was only it was actually 9.7 kb. And he looked at all possible haplotypes of 88 polymorphic sites that they found in that 9.7 kb in a sample of, I think it was a sample of people from Minnesota, there was a sample of North Karelian Finns, and there was a sample of African Americans. And in all of these samples, he did not find the same haplotype of all 88 markers once. Everyone was unique. There was some correlation among these haplotypes of these dinucleotide markers, biallelic markers. There were correlations, there was disequilibrium, okay, within these populations, but there weren't any haplotypes that were represented twice, even in Finns, in this isolate in Finland. So, while there is disequilibrium there, I wouldn't necessarily count on it. And this is looking at disequilibrium among diallelic markers. And, not looking at a disease. A disease is another thing that could occur someplace near here, at which there could be one mutation or multiple mutations. But you don't necessarily expect always to find it unless you happen to have the right population. Now, within Finland, there was strong disequilibrium between most pairs of alleles in the sample, okay? But one thing that I thought that you presented that was the most interesting thing of all is that normally when you think about population history in this model, and you're looking at haplotypes, you're expecting to see something like this, where let's say that the original haplotype, I'm just going to give these different letters at different markers. Let's say that originally it was AAAA. This is 4 alleles. Okay, if you have a mutation at some point that causes this A to become a B, you're now going to have A, B, A, and A in the population, okay? This one goes to here. Let's say another mutation happened at some point to give you A, B, A, okay? Now, you can see that these are possible mutation events, and you can usually take all haplotypes you observe and trace them back and try to find out what was the order in which mutations occurred. Say, for example, I happen to have one here that now gives me B, A, B, A. This one now becomes a B, okay? There are certain combinations of haplotypes that you cannot get, right, very easily from the original one, okay? If this happened first, it's going to be, and this one happened over here, it's going to be very rare to ever see A, B, B, A, because mutations are so rare that most of these dinucleotide, both of these biallelic markers represent single events historically, single mutation events. The same change rarely happens twice in human history. And when it does, it's publishable. That's how rare it is. You can say, we found proof of a recurrent mutation. You can publish that. Although it may be hard to distinguish from gene conversion in practice when it's talking about a single allele. But anyway, you can publish that. Okay? So this, if you see this, it's not consistent with a model in which each mutation occurred once historically, because we know that we have this haplotype and this one, and you can't have all three of those without a recurrent mutation or a recombination event, okay, between this and this. Now, the interesting thing in their studies, we're talking a very small region where you probably didn't have any recombination very recently. You know. This is 9.7 kb, so that's tiny. The haplotypes that he found here, even within Finland, had no such structure. They couldn't trace the cladistic history of the structure to find a single ancestral haplotype that was compatible with what's observed today. So there's obviously a lot of complicated things going on that we haven't completely well understood. But the point is that you do have correlations when you're in the smaller populations between loci, whereas you may not have correlations between a Finn with allele B here and an African American with allele B here. They just may not have, you know, and it may have been so much time since the separation of those two populations that the ancestral haplotype may well have disappeared or not be detected much longer, which is the explanation for why you can see something like this. It's such an ancient event that recombinations have occurred, but you would never expect them to have occurred in the recent enough history of that population, especially when what was really interesting is they've observed, say, the same three haplotypes, both in a sample of Africans and a sample of Finns or something of that nature. So these are ancient haplotypes, anciently originated alleles. Okay? So the punchline of this is, if you're going to do your study, do it in a place with an ethnic group that is homogeneous, where there's a reasonable chance where you can detect some, you know, some similarities between the individuals that are due to ancestral origin of these mutations. Yeah?
This will also depend on the history of the history of
Of course.
mutations.
Of course. That's what I mean, though. This is just one example. This is just one example. You could have millions of examples. The point was that, my only point is, you got to be real careful about it. And in fact, I've done another study. One of the papers that you're handed out was based on a project I did with some population geneticists in Europe. And what we did is we took and looked at, say, well, what happens if we look at a dinucleotide, a diallelic marker, as if it were a disease allele, okay? So this is a case where we're going to just simply look at haplotypes around a diallelic marker, which should behave as a haplotype would around a disease allele, assuming absence of selection or something, for simplicity. Now, what happens is, if you look at a disease and a marker locus nearby each other, and you trace these individuals back to the point at which this specific mutation occurred, because this diallelic marker was created in history, or at least within these individuals, the most recent common ancestor they had, in order to have them today, you find that in populations that have not gone through an expansion, this time of common ancestry is much earlier and much deeper, and it may be so far back that even at recombination fractions of less than 1%, you may not have any residual remnants of the ancestral haplotype. And it was very interesting. We were kind of surprised a little bit by how striking it was. How even if it's only, say, a couple of hundred generations, you may lose all trace stratifications are not that important because you're still going to have strong disequilibrium within the population. But what it means is that if you compare this population to another population of similar structure which broke away at a similar time in history, you'll also have disequilibrium here, but your haplotypes are going to be different, even on the small scale. So it's very important then that you not sample half your people from here and half from here and not stratify on the basis of the population history, because then you're going to not have anything to detect. Yeah? So my point is, to increase the strength of this, make sure that you concentrate on picking the right population to study and don't just pick the one that's easiest to get access to. Because you may not, the thing is, in a linkage study, if there is linkage and you have an infinite sample size, your statistic becomes more and more powerful automatically. Okay? Because there is a correlation there, it's just a small correlation maybe. So by increasing the sample size, you will find something. With an association study, if you have the right population, you may have more power than you do in a linkage study. And if you have a population in which also this correlation is greater, the specific gene's effect on the phenotype is greater for whatever reasons, because of the environment or because of something about that region, that's good, and you'll have a good chance to find it. But if you took, let's say you took and sequenced the genome of the entire world population stratified only on the basis of phenotype, there's a very good chance you don't find anything for breast cancer or for some of these other phenotypes, because there's just so many different population histories that we're mixing as one thing. Yeah? It may well work in simple cases. I know Neil Risch had a paper in Science recently where they advocated association mapping as the wave of the future for human genetic studies. His model was this, basically, that you have some single, the conditions under which his model works most are the conditions where you have a single diallelic mutation in a gene that is affecting the phenotype directly. If that's the case, association analysis, when you have this locus, will work. Also, if you have a marker that is in disequilibrium with this locus, for whatever reason, the study will work. But if you don't have that situation, it may not work, and you may not have that situation in a lot of cases. And that's why it's important to think about population genetics, population history, and getting the right population. Not because, I mean, it's not really a good answer to say, well, decode genetic zones of Iceland's population, because there's also Greenland. And you can say somebody else has Greenland's population. Well, you can say, well, I know there's this other population, there's this Indian tribe nearby, or something like that, where you may be able to find access to it. But whatever the case may be, and I'm not suggesting that that's the only way to do things, I'm just saying that that will increase your power if you can do that. And if you do it with an admixed sample of all different groups, be sure you are very careful and appreciate the dangers. You may find it, if this model holds, where there's a single allele in a coding sequence or in some area around the gene that is the only variant in that gene that's having major effect, then all those analyses work perfectly. Yeah?
Is there a way to, early on in the game, decide whether you are really going the wrong direction?
I think all of these decisions should be made at the very beginning of the study. You should think about these issues before you start collecting people's genotype. Because that's the most expensive part of the studies now, I guess, is ascertaining it. You've got to get your right samples, you've got to collect them, you've got to gather as many variables as you can. I mean, if you're looking at environmental correlations with genes, you want to have all that information too. You want everything up front. So, I mean, if you, and it's sort of something that I know I'm always faced with, and I'm sure you are too, Nick, is people come to you and they say, okay, here's my data, please tell me what it means, where's the gene? And I'm like, well, why don't you have this data, or why did you do this, or why did you do that? And they said, well, we thought we'd worry about that later, or we didn't see the need to, involve someone else to discuss it with at the beginning. And it's true. I mean, it goes back to the thing about, the OJ thing, where they say 6 months on the OJ trial and getting into genetics or something. And you really need to think about these issues to do it right. Yeah. Now, perhaps a more important issue that occurs that's related very much to this is these other things that I had over here. We have genes and we have environments aside from this. We want to minimize as much as possible those effects. How do you minimize the effects of these guys? That's very complicated. I don't know that we know how to do it, with the exception that in these smaller populations, as was mentioned in the context of these markers, alleles are more likely to be fixed and not so variable. So if, for example, you were looking at group of Siberian herders or something who didn't have very many, who had been a very small population, there's a very good chance that at many of the loci, if there was, let's say that there were 6 or 7 loci involved in the phenotype, they may be fixed at 3 or 4 of them. They may have no variability. That increases your power, of course, to detect other loci, assuming that there's some correlation between them. Because you don't have to look for all of them. You don't have noise from all of them. You have less heterogeneity. Yeah? Everybody who's got the effect is probably sharing, are more likely to share the same genes than they are to share different genes. Now, but aside from that, it's very difficult to control for this, except through picking people who are related, who have similar ethnic background, et cetera, et cetera. The environment, though, that's something, environment and gene-by-environment interaction are things that are somewhat more amenable to analysis, either through the proper ascertainment of samples or through collection of data at the very beginning about these environmental factors which are hypothesized to be involved. There's a long tradition, I think, in epidemiology of treating genetics as something to be integrated out of the analysis. It's just noise, and we treat it as some normally distributed garbage that will just sort of figure its way out in the end. Geneticists are just as guilty. We take the environment and we integrate it out. We say it's normal. And it's equal for everybody with possible correlations. The more sophisticated analyses say, well, people are related, so they share something more likely than not. So they have their own normal distribution for the similarity factor. Yeah? That modifies the mean of the distribution somewhat. But it's not that simple, right? So if you have the data, you use it. Yeah? If you're looking at genetics of lung cancer and you don't collect whether or not they smoke, your study's not going to be very wisely designed. Yeah? And we can factor this information in various ways. There are statistical ways to deal with it. But perhaps the most important thing is to try to do something at ascertainment time about this stuff. Yeah? If you want to look at, people who have really high cholesterol but never eat fatty foods or something, that may be more interesting than looking at, people who eat bacon for breakfast and a pound of bacon a day or something, and they have high cholesterol. Yeah? Well, I mean, it's a very important factor, because also eating bacon for breakfast segregates in families, because mom cooks sometimes for everybody, right? So, I mean, there are real reasons to think about these issues at the time you're collecting the data. Now, I mean, I think that that's very important, and that also gets to this. This is very correlated with this gene-to-phenotype gene-to-phenotype reaction. If you have something that's rare, if you want something that's rare, we can apply simple techniques of linkage analysis, simple techniques of association analysis, and we can map those genes. But the ways to do that are through finding people who have all the environmental, things that would give you the disease for various other reasons, and you get rid of them. Like, you look at guys who got breast cancer. I mean, that's not environment, that's other genes, I guess, right? That makes you a woman. But, I mean, if you still get the breast cancer even without that, then that's a more severe correlation, and that's a useful thing to do. Anything odd is good. Weird is always good in genetics. That's why we're all the way we are. Okay, so anyway, this is all dealing with the ascertainment issue, which I think is the most important issue that you can discuss. Okay? In any of these studies, the most important thing is this. The analyses are trivial. Analysis is simple, okay? It's all basically the same. We tend to make it a lot more complicated than it is because, well, we have to. If we don't make it more complicated, no one will use our programs, and then we don't have careers. No, seriously though. I mean, it's basically pretty simple. To think about it, let's go back to what I started talking about a little bit before. The idea is this. In the simplest study, we want to see, is there an effect of a gene on a trait? Let's start with an association analysis, the very simplest case. We have our cases and our controls, and we compare them. Is there a difference in the frequency of allele whatever at our marker locus among affecteds and unaffecteds? The reason we do that, these guys are enriched for the disease allele or alleles. These guys are enriched for those that do not have the disease. Now, there's going to be some disease alleles here, and there's going to be some non-disease alleles over here. We're just trying to maximize the difference between these two groups in our ascertainment. Okay? You want cases and controls. Controls may even be a bad word. It's sort of to say you want affecteds and unaffecteds, where unaffecteds could be even selected to be beyond just unaffected. They could be someone who's the opposite of affected. You want to make these phenotypic groups as different as possible, so that these guys have disease alleles, these guys have wild type alleles. Okay, you want to enrich for that difference, and if you find a significant difference in the frequencies of alleles between those two groups, that's evidence of some correlation. Yeah? And that would be looking at this thing we were talking about before. That's the simplest kind of analysis to do, right? This is just a this is just an epidemiological risk factor, nothing more. It's trivial, very simple. The alleles at the marker locus are a proxy for a risk factor. You can apply your whatever test you want. You can do a likelihood ratio test. You can do a 2x2 chi-square test or a 2x n chi-square test if there are multiple alleles here. The analysis itself is simple, but this is what you're doing. You're contrasting these two. So let's say that, for simplicity, let's say we were doing something with likelihoods and we were just going to compute what's the, we'll estimate haplotype frequencies here. We have allele 1 and allele 2 at the marker locus. And let's say that here we're trying to determine what's the probability of one allele given we're affected, right? Why do we need our controls? Because we need something to compare this to. That's the only reason we need the controls. Because we don't know. If we just compute this and probability of 2 given disease, there's nothing here that tells us anything unless we know what it is in the wild type population. So we collect our controls basically to get the allele frequencies, P1, P2, or if we've ascertained these in such a way as to make these even more unaffected, then this may be P of 1 given not affected, or given this extreme other phenotype, right? So we just want to find out, are those equal? That's the whole test. Yeah? Very simple. I won't get into the statistical formulas because it's kind of not so relevant. The most important thing is that you see what you're comparing. Now,
Can I just interrupt? At this point is a question of ascertainment. Is it important to select these unaffected from the general population?
The most important thing here is that they're genetically independent samples, that every case and every control are genetically independent, and that for a marker that is uncorrelated with the disease, they should all be IID samples, independent and identically distributed. Meaning, if we have an unlinked marker, these guys cannot be related, and they have to be from the same genetic background. And it's a very difficult thing to do in practice. I've been involved recently in a study in an extremely isolated population where they collected a sample of affected individuals and a sample of unaffected individuals, and they tried very hard to make sure that they were approximately the same genetic relatedness. Well, they weren't. And as a result, you found a lot of false positive evidence for linkage disequilibrium throughout the genome. Because the fact is, and it's a very important fact, when you're ascertaining cases and controls from an isolated population especially, it's almost impossible that the cases aren't more related to each other than the controls are, no matter what the genetic model you assume. Because, we're not looking at something here that's 50% of the population. We're talking about a phenotype that might be 10% prevalent. Well, they're going to have some genes in common with higher probability than anybody else. If they have genes in common, they're more likely to be related less distantly than other people are. So it's something you do have to be aware of. What I normally would say is that in these kind of studies, one very simple approach is do the whole genome. If you're planning to do a genome scan, do your genome scan and use the built-in empirical controls that you have. If you see something that's way, way, way more significant than you have anywhere else, that's pretty interesting. But if you see something that you would normally call a significant test, let's say you've got a p-value of 10 to the minus fifth, but you see that same thing 10 other places in the genome, I'm betting you don't find 10 genes with this kind of study, yeah? In your initial screen. You're not going to have that, probably. If you do, you're incredibly lucky, and you'll be instantly, the most famous person in the world, because it hasn't happened yet in genetics. These days, if you find genes for rare recessive diseases, you can get on Dateline NBC like happened last week with this baldness thing. So, if you could do this,
So randomly population selected unaffected would be much better than maybe unaffected cases of pedigree siblings?
If you're doing a case control study, I would say that unaffected who are extremely different in the phenotype may be better if you have a match for the genetic population background. Because they're more like, you're not trying to compare it necessarily to the whole population, but to the opposite. You want to contrast these groups. What you want is samples that are as different as possible when you are near the gene, but as similar as possible when you're not near the gene. Okay? You're not really trying to compute a relative risk, if you will. You're trying to maximize the discrimination between those two groups based on one or a small number of genes. That's always the goal here. We don't really care about the epidemiological parameters because we're not ascertaining in any well-defined strategy. Nobody can ascertain. And even if they say they're going to ascertain pedigrees using single ascertainment, if they happen to see a family somewhere that has a couple of sibs or a couple of relatives, they're going to jump on it. That's wise, you know? And so nobody follows a rigorous enough ascertainment scheme you can actually correct for it appropriately, so we don't worry about it, and we assume that all of our parameters are going to be misestimated if we try to quantify relative risks or various things of that nature. Okay? Now, one control way that people deal with this problem, and what you just mentioned, is they use the parents. If you ascertain a pedigree of this structure, yeah, and let's say at the marker locus we have 1, 2, 3, 4, and 1, 3. We can use 1, 3 as our case genotype, and 2, 4 as a random genotype from the control population. This is the haplotype relative risk design that Falk and Rubinstein proposed in the late 70s. And it works very well. But there are disadvantages. There are advantages and disadvantages to this strategy. Obvious disadvantages: here you have to do 3 genotypes, here you have to do 2 genotypes. From these 3 genotypes, the power is less than for those 2 genotypes if everything is well controlled. Okay? So depending on what your goals are, there are reasons why I would do this study first. I mean, if my goal is to do a genome scan, this is not going to miss positive results that this will detect. This will detect too many false positives. Okay? Because if your samples are not well matched, you probably are tending to find positives everywhere. Okay? This will have many fewer false positives. That's a reason to do this and then follow it up with this if you find something, I mean, it's a cheap way to do it. I'm only thinking here about economics. If you can do, 50% more samples this way than you can this way because you're doing, 1 extra genotype here, I would try that just to increase my power. Perhaps it depends on the situation and how confident you are that these are going to be reasonably well matched.
We could have another control, more than one control.
You can, but I'm saying that's exactly what I'm saying. I'm saying that you would do this, and if you can get the parents, you get the parents, and you do this also. But a big problem is that most complex diseases are diseases of late age of onset, in which case the parents are usually dead, and it makes that very difficult to do this. In one of these papers that you got handed out, I've thought about ways of doing this kind of a study when the parents are dead, and doing a case control and this in one analysis. Just computing the likelihood of the data. It's one of these papers that was, I guess, handed out or will be handed out. Oh, you just got this one. Okay, it's going to be at some point. Yeah. Yeah, the paper that you have there describes other mechanisms for the generation of linkage disequilibrium. I'll mention that if there's time when I finish this. The bottom line in what's in the paper that you have handed out is that there are reasons why high mutation rate of a marker locus is good for association studies, because it can generate disequilibrium in populations that are really small. If the population is really small, mutations affect disease and plus chromosomes differently, so you can increase the difference between the two groups. I'll talk about that if we have any time later. I want to get through some of this stuff now because this is probably more important. This you can always read about. But anyway, this is what the difference is, okay, between these two basic approaches. Yeah? Now, there's something that people do a lot, which is a variant of this called the TDT test. Now, what's the difference? The difference between haplotype relative risk and TDT has to do with this kind of individual. You have a homozygous parent here. Well, if you're interested in looking at linkage, if you're thinking about this one in a linkage context, that's uninformative, right? Because you can't tell, you automatically have a 3 transmitted and a 3 not transmitted. But in a linkage disequilibrium context, there actually is some information coming from that. if you're ascertaining singleton individuals and their parents, there's information there, okay? This is the haplotype relative risk versus as compared to a TDT testing. We studied this in a paper I did with, when I was a PhD student with your guy, and we called the TDT a McNemmer test. It's basically a paired sampling test. And we looked at it and concluded that for basic linkage disequilibrium analysis, it wasn't very useful. It gave almost the same power as haplotype relative risk, but it was a little bit less. The only conditions in which the TDT is actually better than the haplotype relative risk are when you have a huge deviation from Hardy-Weinberg equilibrium, meaning that you have like great inbreeding in your data or something of that nature. Or you may have population stratification. But if you have population stratification, why would you have linkage disequilibrium? That's the question. If you have, say, half your sample was whites and half your sample was Japanese, why would the same allele be associated in both groups with the phenotype? Unless it's this very simple model that Neil Risch was talking about in his study, where you have one mutation in the gene that is the disease allele and one wild type. Well, the wild type could be whatever, but it's only one mutation in all populations causing the disease. Now, there are going to be cases where that's true. I just doubt that that's the case on average. But, you know, people, we all have our different opinions about it, and I don't want to say that what I'm saying is right, because I'm not saying that. Nobody knows. But that's always been why I haven't used the TDT very much, because it doesn't necessarily, it doesn't seem logical that you would have such extreme problems with Hardy-Weinberg and still have disequilibrium. And if there is Hardy-Weinberg disequilibrium, it means you have too many homozygotes, yeah? Not too many heterozygotes. These are basically not going to increase your false positive rate. They're going to be decreasing it. In fact, you're going to lose power because you're adding a sample to both case and control, which are identical. So you're homogenizing the results. So it doesn't really hurt. It doesn't increase the chance of making type 1. And anyway, this is basically the main strategic design association study. Okay? Now, in a linkage study, I'm going to claim that there's no difference between any of the studies we do and the logic of the case control study. And I don't think a TDT is any different either. Okay? The only difference in the TDT is that you're genotyping the parents. You're using more information. Here, you have just your disease individuals, and you have your sample of controls, which you're using to estimate the normal allele frequencies. In the TDT, you're genotyping the parents, so you have more information there, and you no longer need these guys because they don't contribute any additional information. But you can use them anyway if you want to. You can just add them into the data and do the analysis together. But here, the parents are genotyped, so you don't need the control. Now, we add an extra sibling. We're now in the case where we're doing some linkage analysis. We can also do association analysis. This is the same as a TDT pedigree, except remember, we have one, if we assume rare recessive disease for simplicity at the moment, what we're going to have, let's assume that this guy's uninformative. It doesn't really matter what his disease locus. But here, we have one haplotype that we can estimate. There's one observation for disequilibrium, or two at most, the two parents. Okay? That doesn't change when I add more SIDs. There's still only those two individuals who are random sampled from the population. These guys are going to be correlated due to linkage. But this and this pedigree, any correlation in the marker locus genotype across pedigrees is only going to be through the parents, the unrelated individuals from the population. So there is, of course, linkage disequilibrium information available in any pedigree study. Yeah? But there's one observation that's contributing to the haplotype frequencies. In the TDT test, there's a great habit of looking at all of these four as if they're independent samples. Okay? Treating them as four individual cases from the population, but they're correlated. So it's not really right if your test is there disequilibrium. It's perfectly right if your test is there linkage. TDT is designed to test linkage. That's why they can use multiple SIBs from the same SIB shape. But if there's no disequilibrium, there's no power. Okay? But a positive TDT in multiplex pedigrees tells you nothing about linkage disequilibrium. You do not have to have linkage disequilibrium for this to deviate from its null hypothesis distribution if you have large pedigrees. Okay? In this other paper that you'll be getting at some point, I did give you a simulation result for a couple of cases, which clearly shows that how the TDT becomes increasingly non-power, increasingly anti-conservative when there actually is no disequilibrium, but your pedigrees are big. Yeah? It's something that's underappreciated. If you see TDT, it doesn't mean disequilibrium. It means there's definitely linkage, and if there's singletons, it means that there's disequilibrium. It can reject the hypothesis of no disequilibrium only in that context. But now, back to the point. Why are these basically the same test as a case control test? Well, if I genotype the parents, much like in the TDT, and I type the kids, all my linkage information is there. I don't need a control sample, right? It's much like in the TDT case. I can see, is there correlation between these SIBs at their genotype? That's linkage analysis, like I did before a few examples of SIB parent tests. Now, if the parents aren't typed, however, all of a sudden, I need a control sample. We don't think of it that way, but that's what it is. This is doing the same function as it's doing here. It's giving us the null hypothesis distribution for our alleles in the pedigree. If we're only testing linkage, we normally don't care whether this one's 1, 2, or 3, 4, yeah? If everybody's genotyped, it doesn't matter. But as soon as this person's not genotyped, we have to guess at their genotype from the genotype probabilities, given allele frequency estimates. Those allele frequency estimates are obtained from a control sample. Where else do you get them from? If you assume wrong allele frequency estimates, you get the wrong answer. You get false positive evidence of linkage. So it's the same thing. There's no difference. It's just that you're collecting larger samples. Yeah?
If you have enough siblings, can you infer parental genotypes?
The more siblings you have, the less relevant it is, the parental genotypes. But let's say that you had 10 siblings in a SIBship, and they all happen to have genotype 2-2 at the marker locus. Okay? Let's say you had 100 siblings that were all 2-2 at the marker locus. All of a sudden, your parents seem probably to be homozygous, and therefore that there's no information. But what if the allele frequency of the 2 allele was 1 in 500,000? All of a sudden, it looks like you got linkage, right? So there's always going to be some reason. If you're always dependent on your genotype information that's available to reconstruct the parental genotypes, you're going to tend to bias yourself against linkage. Because you can only reconstruct the parents when you observe all the alleles. And you only observe all the alleles. If you're only typing affected children, you're only going to observe all the alleles in cases where there's discordance among the children. Whereas if they're concordant, you're going to assume there's a greater chance the parents are homozygous. So it is important. Yeah. So the point is really that you're always doing something that's almost the same as a case control study. If I compute the likelihood of the data here, it's no more different than computing the likelihood of the data here, just that here I suddenly have the recombination fraction as part of the likelihood because I'm looking at linkage. Now, this is always true. In any linkage test that you do, where the parents aren't typed, you need a control sample. The only reason that you type it is so you can determine, are these cases, the same thing here, more different than I would expect if there was no linkage? I don't know what I expect if there's no linkage unless I know those probabilities. And the only way I know those probabilities is through a control sample and or through genotyping other individuals in the pedigree who aren't themselves affected. Okay. Now, another common source of discussion in these linkage tests is sort of the idea that there's two classes of tests. There's parametric analysis and non-parametric. These are kind of, in some ways, not necessarily the best definitions, I guess. I got someone, a reviewer in a grant I recently proposed, complained that I used the word non-parametric, which I obviously didn't understand, therefore I needed to go back and take basic statistics again. Which seems a little strange because I was just quoting the literature when I said it, you know, calling SIB pair tests non-parametric. But anyway, he preferred that these would be called model-free and these model-based. But that's not really good either, because in some way they're all model-free and model-based. But I think there's a lot of misunderstanding about what really does it mean, okay? Normally we think parametric analysis means you have to specify a model, and that model is always wrong, therefore it's not a good thing to do. Non-parametric analysis, we don't have to make any assumptions, therefore it's a good thing to do. But what are the assumptions? Now, I was sort of trying to figure out all of this in my head because I was always trying to make sense of, well, why is it that sometimes sometimes when you submit papers and you don't do the SIB pair analysis, you'll get a review back saying, why don't you do non-parametric methods? Parametric methods are known to have all these problems. And then if you do the non-parametric methods only and someone else reviews, they say, why didn't you do linkage analysis? We know that it works. We don't understand all these non-parametric methods, or what do they mean? The thing is that there's a very simple difference, actually. I tried to figure this out, and I went back to the very first principles of what we're doing, okay? Now, what really is the difference between a parametric and a non-parametric analysis in the most cases? Well, if I take, let's say I have a pedigree, let's say I have a SIB pair or something, okay? Now, if I do the analysis and I say the parents are, let's say those are their genotypes, and these have some genotypes of their own. What am I assuming if I do a SIB pair analysis? And I say, I don't want to assume anything at all about the genotypes of the parents. I don't want to assume anything about the genotype-phenotype relationship. Well, the way almost every analysis handles this is they assume every meiosis is IID, and they're all informative at the disease levels. What they're basically assuming, in the case of SIB pair analysis, the SIB pair mean test is sort of assuming that both parents are informative at the trait locus, because they're assuming that the probability that they share an allele from this parent and from that parent are identical. That's the basis of the SIB pair mean test. It assumes that you have an equal probability of sharing any allele from either parent. Now, if we have a relative pair analysis, where we have cousins, what do we have? Well, there's only one way these guys can share an allele, and that is through this path. And one of those two parents being D, having this D, and the other one we don't care about, because we call it plus plus, but it doesn't matter what it is. The fact is there has to be a single disease allele transmitted that way, is the only way they could share an allele identically by descent. When we do the analysis, and we want to compare different types of relatives, we assume that every meiosis through which the disease segregates is independent and identically distributed. Okay? This is the basis of model-free analysis. We don't usually talk about it that way, but it really is the case that we're assuming something. We're not assuming something. We're assuming that we're just going to say every meiosis has the same distribution, or every cousin pair has the same distribution, and every SIB pair has their own same distribution. But we're assuming something there. That something is very equivalent to a lot of things we do in parametric analysis. It's kind of like picking a model in which every meiosis is maximally informative. Now, what is parametric analysis? What does it mean if we specify a model? Well, let's say, for example, this parent here is affected. If we do what we call non-parametric SIB pair analysis or something, we disregard that information. We're not using all the information that we have because we don't want to make assumptions about the disease locus, the genotype-phenotype correlation on that level. But what do we do in parametric analysis? We use this to determine the probability that the meiosis are informative. So we're sort of fixing a model which fixes a structure to the pedigree, which says, if I say the penetrance here, let's say I pick a disease allele frequency that's rare, so it only comes in once, but let's say I assume it's dominant. Well, if I assume it's dominant and there's no phenocopies, it's going to assume that this guy is D plus and these guys are D plus. So it's going to say, let's ignore meiosis from that parent because that parent's going to be plus plus with the trait locus because they're unaffected. Okay? So it's directing your meiosis in a specific predefined manner. That's what assuming a model does. Okay? It directs you a priori to deciding which meiosis are informative, whereas in non-parametric analyses, we assume they're all IID, but that a fraction of meiosis are not informative. Those are the unlinked ones or whatever. If we're doing SIB pair analysis and 70% of alleles are shared, we're basically doing a heterogeneity analysis where we're saying we do this kind of thing, and we say there's a certain percentage of meioses that are uninformative, but it's equally likely that any one of the meioses is that meiosis that's uninformative. Okay? Whereas in a parametric analysis, we predetermine which ones are informative, or we at least assign prior probabilities to them. It's kind of like the difference, in some sense, between, it's really a difference between how much you want to specify, how much information do you want to use. And it's basically how reliable is this information. If we're doing a complex genetics analysis of complex disease, we may not want to have to make the assumptions about the mode of inheritance. So we assume every meiosis is the same, and we'll see what washes out. We have a heterogeneity parameter, meaning a percentage of meioses are not going to be segregated. That's really the only difference between the two. Which meioses are informative? And I think if you think about it enough at that level, we realize that there really isn't a great schism in linkage analysis. These people that want one type of method versus the other are not really arguing about something that means much. They're just arguing about, do I want you to specify the mode of inheritance, or do I want you to estimate it in some context, or estimate the proportion of a meiosis in which there is linkage, if you want. Yeah, there was some, yeah.
Yeah, if you had, where you had those cousin pairs there, suppose that originally grandparents already, one of those wasn't affected.
Okay.
In that case,
Yeah.
If you had that information, you know, you had good data that said that even those people were probably deceased, okay? So now, which would be, and let's say you had several samples like this, okay? Now, let's say that the only biological samples you could get the genotype would be in the younger generation.
Right.
You didn't know without the parents.
So these two are both dead, and one of them's affected.
Right, but you knew one of them was affected. And what would be the most powerful to do? Would they just do the real pair analysis, or actually do the
Most powerful analysis is not possible to define. If it was, nobody would argue about it, and there's millions of arguments in the field about it. I think in a large part, there isn't going to be a huge amount of difference. It depends on what the true state of nature is. I mean, in a model-free analysis, really you don't use that information because you're not trying to assume anything about the meiosis. Right? And you're assuming they're all the same, and since this contributes no information, you know their genotype, you're not going to use it unless there was some way that a reconstruction helped. In that context, it won't in this case, but in some cases it might. I don't really, so what I want to say is I don't really think there's a difference between the relative pair approach and the parametric approach, with the exception that in the relative pair approach, you're in some context overestimating the strength of the genetic model, but you're compensating for it by not caring about theta, if you will. You don't care about the estimates of your parameters so much, but you're willing to say, I'm going to assume a pretty strong model in which every meiosis is independent and identically distributed relative to the informativeness of that meiosis for detecting the combinations. Yeah.
It looks to me, is there something relationship there? I remember having read in some of the papers of somebody sitting here that one of them is said based on rejecting a model, and the other is really fitting a model as you've been fitting the parameters.
Well, yeah, you can fit a model a priori in a parametric analysis, and then if you find no evidence of linkage, it means if that model was correct, there's no evidence of linkage. And if you're doing one where you're not specifying a model, you're saying that there's no correlation between their genotypes. Yeah. There are cases where this is better, and there are cases where this is better, but you can't know a priori because you don't know what the true state of nature is. The question is really, I mean, in all of this study, the null hypothesis is never defined very well in a linkage study. What is the null hypothesis in the genome scan? That there is no gene, or that there is a gene of unknown location, or, I mean, are you trying to, I mean, it's a very, I mean, if you get into the philosophy of what you, the way you just put it, it's very complicated. I think we end up thinking that we don't really have a null hypothesis, or nobody wants to believe that the null hypothesis should be there is no gene. Because if that's the case, then these aren't the right analyses to do because we're not using segregation information and ascertainment corrections or all of that sort of thing.
That is just a quantum philosophy.
Yeah, exactly. And I try to avoid that kind of stuff and rather just look at what's really basic about it, you know. if we're thinking about detecting a gene at a specific point. I mean, that's really the difference. It's sort of like, in a sense, if you think about it, SIB-pair analysis here, in a very simple way, is the same thing as a linkage analysis, assuming something that is recessive, because you're looking at both meioses as informative and which both parents are heterozygotes. However, we don't like theta, so what we're really doing, which is also mentioned a little in this other paper, is we're taking each marker that's linked, and we're saying that theta actually goes in this direction, it doesn't go along the map. Because the idea is that if you had a theta of 30% with this marker and 30% with that marker and 30% with that in a two-point analysis, and you tried to look at them together, you'd be saying the disease is nowhere because these estimates are never consistent with any position if you tried to use this recessive equivalence. But if you put the recombination fraction in a second dimension, it works, and it's consistent. And it allows multi-point analysis to work in parametric and non-parametric methods by the same comparison, because you can do your multi-point analysis as really a two-point analysis versus the IVD sharing at some perfectly, maximally informative marker at this position. So it's kind of saying, because our model's wrong, the disease is pushed away in some error direction, this being the percentage of SIB pairs that that don't share the allele IVD, or it being the recombination fraction under a specified model, because they mean that model's wrong. And in complex disease analysis, every model is wrong because you can't do it right. So we don't even, worrying about whether the model's right or not is kind of a moot point because it's wrong. In fact, it's wrong for simple genetic models too. It's wrong for cystic fibrosis because it's not simple, rare, recessive disease where we know the allele frequency and everything else. It's just a guess. It's a pretty good guess there, and a pretty bad guess in a lot of other traits. But that's basically the point.
Joe?
Yeah.
We're into our discussion section.
Okay.
Did you have any key points that you wanted to wrap up from your presentation, and then we can move into?
Okay, sure. I mean, basically, what I would like to say is, I don't necessarily think it's important to dwell on the differences between these different linkage techniques or the different association techniques. They each have their good points and their bad points, and none of them is optimal. They're all actually variants of a case control study in some sense, because you're always taking a control sample to recreate the null hypothesis distribution. You want the null hypothesis likelihood. You can't get that without some controls, or be it parental genotypes or genotypes of the whole pedigree and context. You always are doing the same thing, really. Whether it's a likelihood approach or a chi-square approach or whatever, you're always trying to compare the distribution of your data given linkage versus that given no linkage, and looking at the difference between the two. So there really shouldn't be the great isms that there are in the field about which test is better than which test. It's really largely political posturing, because everybody's model is based on their own theory of what reality is. We know reality is more complicated than any model that anybody ever has simulated in any study ever to predict the power of any method. Every grant overestimates its power probably 1,000-fold. Everything is done to inflate the significance. So there's no sense in arguing about which is better, because we can't. We don't have the data. When somebody starts finding these genes, we'll be able to do something about predicting it. When it comes to ascertaining things, always try to ascertain in ways that you maximize the correlation between markers and disease, be it through increasing numbers of markers or increasing the type of population, the isolation of the population. If it's an association study, you want to get people that are as related as possible, as ethnically homogeneous as you can, because they're more likely to have the same gene. These are only ways to increase the odds. There's no way that anyone can give you the odds. We can't simulate something and give you the probability of finding something, because we don't know what reality is, okay? So any simulation anybody ever does for you will be too optimistic, and the powers are only meaningful because you have to convince people on grant committees that you understand that there's a problem and that you do need to have an idea of the power. But always try to do whatever you can to increase that power, and don't worry too much about the formal algebraic details, because those we have computer programs that are very easy to use usually, and they're becoming more and more easy to use. And certainly before you start a study, you should give Nick a call, or somebody in someone of us, so we can maybe give you guidelines for getting through these things better. A priori, though, don't wait till it's too late. Is there any questions? Yeah, that's it. Okay.
Thank you.
I thought we'd start first with questions. conversation started, whether about particular projects that investigators are working on in the CEG, some questions about, some general questions about study design, issues that sort of have initially gotten raised here that you wanted to hear a little bit more about.
Joe, you had this alluded to that for disequilibrium studies, markers with high mutation.
Okay.
Yeah. Microsatellites.
Okay, as I understood, he was asking about something I mentioned briefly about marker mutation rate and disequilibrium. What I said is a little bit different, I think, from what Nick said this morning, only because I'm looking at something, I'm thinking about something different than he was thinking about it. Can I? It's okay.
And also, usually S&P, with large.
Right.
So what's going to be, the choice of markers, in complex disease?
Yeah. I can say what I did is in the manuscript that I just had handed out is going to be in human heredity next month, and we were trying to look at that question a little bit differently. Our model was this, that if you treat disease alleles and plus alleles as if they were two basically independently evolving populations, as they are, right? These are passed down from generation to generation, but there's no guarantee that there was a founder effect of any kind, or there may have been, but we don't know. Now, if you're using diallelic markers, and a whole bunch of them, the only things that are going to affect the distribution of any given allele, basically linkage disequilibrium means that there's a difference in the allele frequencies of a marker locus in this population and this population, right? Populations of chromosomes. Now, mutation rates, if the population is small, if this total is a small group, then mutations that occur independently in these two groups are going to increase the differences between those two groups, yeah? A mutation that occurs here at a marker very close to the disease allele is going to create a further difference between the disease E population of chromosomes and the plus population in which that mutation did not occur. Now, if you have a huge population where the microsatellite alleles are in some sort of an equilibrium, that doesn't make any effect on the locus at all, right? It will decay. I mean, obviously, if you're looking at a model where you have a single haplotype of these diallelic markers, recombination is going to decay it, and mutation is going to make that haplotype go away. But if you're dealing with populations where the disease allele is fairly common, and where the whole population is small, a mutation rate at a microsatellite can create disequilibrium, meaning that the allele frequencies at a set of marker loci nearby to the disease allele will become more and more different in this population than this one, although there won't be haplotypes. Because they will dissolve haplotypes, but haplotypes and disequilibrium are not the same thing, okay? Haplotypes are disequilibrium only if you're talking about an ancestral chromosome and its decay. So I don't like SNPs. I think personally that they're not so good because there's another reason. Mutation rate is going to, I've never seen the population yet, no matter how small it is, in which microsatellites don't have some polymorphism. But SNPs are often fixed in these small populations. So if you want to do a linkage study in a small population, the SNP map that's being developed is being developed on probably the most heterogeneous populations in the world. I mean, they're taking admixed groups and trying to find something that's polymorphic there. It's not gonna be polymorphic in, Inuit or something, you know?
That's what I was wondering about the recent search on the identification of SNPs.
Yeah.
So much of the importance and good, I don't know that, whether SNPs, I'm not a mathematician, whether SNPs are going to be more useful in mixed populations.
There are arguments for and against them, I think. Another argument against them is that you can't estimate haplotype frequencies between markers or allele frequencies at markers very efficiently, or genetic distance between markers. Physical distance they can get, but the physical to genetic distance hasn't been very good on the small scale, at least according to Lynn Jordy's stuff that he did with the disequilibrium. So if you don't know the genetic distances and you don't know the marker allele frequencies, your analysis is making many, many, many more wrong fixed assumptions, and it's going to lose some power as a result. I think I'm probably one of the more pessimistic about them. I think Nick is more in favor of them, so I think he has to be. I don't have them, so I don't have to worry.
follow up on that. Joe's right in that there are advantages and disadvantages, and I think one of the reasons we all thought having someone like Suda around today would be important is because you could describe some of the advantages and disadvantages of these markers. SNPs are found with greater frequency across the genome. That might be an advantage. You get closer to the actual disease mutation. Admittedly, that might cause problems when it comes time to do the disequilibrium analysis because physical distance and genetic distance might break down. But you could think of it as an SNP might be more likely to actually have some functional effect because of just the nature of the mutations and repeats, although they've been found to be implicated in certain diseases, they by and large probably aren't contributing in a substantial way to lots of diseases. So there are advantages and disadvantages. Plus, lots of SNPs may, and you've been written about this, lots of SNPs could produce the same sort of informativity as one microsatellite in terms of the number of alleles. But there are issues even in that analysis.
Yeah.
But the number of SNPs would have to be because of the because the informativeness of SNPs is lower than.
Okay. And then we could get into, Joe had mentioned sort of the economic basis of this. Maybe it's just easier to genotype SNPs in an era of gene chips. So 50 SNPs might be as good as one microsatellite, but if you can genotype 50 SNPs at half the cost as one microsatellite.
Yeah, sure. There's somebody, I don't know who told me that they had heard some rationale the SNPs were only being proposed because they couldn't do the sequencing of the genome without them, and that was just an excuse the linkage mapping.
Well, they're sort of also a byproduct of sequencing.
Yeah.
Yeah.
You're going to discover them by default. Yeah. So we might as well try to make use of them.
Yeah.
Additional questions?
I reckon, Nick, that when you gave the seminar a couple months ago, you talked about the approach that you get proposed, provided you had a dense enough map that the environmental that the disease gene that you're looking for might not have to be 75%, but only 25%, and the other factors contribute. You can still find it. Okay, so you want to make any comments on that, Ralph?
Sure. Well, again, what I was getting at there was the relative power of a linkage analysis versus an association analysis. And I think maybe I could give one brief example on the chalkboard to show why statistically linkage analysis is less powerful. And it's just one example, and it's probably raised more questions than it answers, but it's an example that I've given to students and had them think about. Simple nuclear family, these two individuals are affected. These are the genotypes of the parents and the genotypes of the offspring. Now, one statistic that's used often to quantify the number of alleles shared by these SIBs is the number of alleles shared identical by state or identical by descent. Okay? And what we want to do, as Joe pointed out, is somehow compare the number of times we see siblings share 2, 0, and 1 alleles identical by state or identical by descent with what we would expect if there was no association between a particular allele, marker allele, and the disease. Okay? Now in this example, we'd have a heck of a time coming up with just how many alleles the siblings are sharing identical by descent. We don't know if in fact they're sharing 1, 2, or 0 alleles.
0 or 2.
0 or 2, I'm sorry. So there's some ambiguity there. But if in fact this is the disease locus, these alleles are somehow associated with the disease, we don't care because they have genotypes that confer the risk. Okay? So somehow the information that we're getting out of this statistic has ambiguities built into it that we don't really care about. In other words, why would we care about the origin of the alleles here? Whether they're coming from the one parent or the opposite parent. We don't care. In other words, we don't care about how many alleles they share identical by descent. We care about what their genotypes are at the disease locus. In other words, if the 2 allele was dominant to the one allele. And it caused disease 100% of the time, so it's dominant, fully penetrant. All we know is these two individuals have disease-predisposing genotypes. Okay? Do we really care if they're sharing 0 or 2 alleles identical by descent? There's an ambiguity in that test statistic that we ultimately don't care about. If, in fact, this was the disease locus. Now, for association analyses, what we're trying to do is just see if, in fact, individuals with certain genotypes have the disease more often. Linkage statistics, because they make use of identity by descent information at times, suffer from having this built-in ambiguity that is sort of irrelevant to identity by state for just association analyses. Okay? So, if one wanted to know why there's a certain lack of power associated with linkage analyses from a statistical sense, you could argue that some statistics have built into them ambiguities that you don't really care about. When in fact there is a direct association between alleles, that is, you're at the disease locus.
How would you test that with this data?
You wouldn't necessarily. All I'm saying is if you wanted to go ahead, if you wanted to look at an association between particular genotypes and disease, what you find is these individuals and other individuals that have the heterozygous genotype had the disease. You'd see a good association if in fact there was a disease gene, this 2 allele, that caused disease unequivocally and 100% of the time. Okay? So why would you want to make use of information on where the origin of the alleles, whether they're shared identical by descent or not? You wouldn't necessarily want to. And yet linkage statistics rely at times on information like that. So again, with some statistics used to quantify linkage, there's ambiguities in them, namely the number of alleles shared identical by descent, that you really don't care about if in fact you're interested in just doing an association between the genotypes and the disease. So in fact, if you did association analysis and you were at the disease locus, you're not next door, you're at them, then obviously you'd have greater power because that analysis would not suffer from those sort of ambiguities in the number of genes shared IBD. So the point I had made in a couple months ago is if you could do an association study throughout the entire genome, and that we're probably limited by the number of markers, then there's good statistical reasons why you have greater power. That in some of the linkage statistics, you're biased against having a good power because of these built-in ambiguities in the sort of statistics you're relying on. That is, we don't really care about the origin of the alleles and the number of alleles shared identical by descent, if in fact you're at the disease locus. Okay? So I actually went through a little statistical exercise to show what kind of loss of power you have in doing straightforward association tests with, or as compared to a linkage test. And they sort of bore on in these sort of ambiguities and the reason why these ambiguities go out the window when you do an association study. Okay. But let me just say, just because I'm going to have to leave soon, there's one thing that I fully agree with Joe, and that is that a lot of these genome scan efforts are truly empirical exercises. I mean, this is one thing that it goes without saying. I've been involved in debates with people I've collaborated on before about criteria for declaring significance, and a number of those debates, I think, were sort of ill-conceived because everybody knows these things, or everyone should know that these exercises are truly empirical. We don't know the complexity of the disease we want to study. We just want to go ahead and do the study and see what emerges as a result, and then go from there. Okay? So designing studies, it is important to pay attention to all the things that Joe had mentioned, that I had mentioned. But ultimately, assuming that there's some very strict and rigorous null hypothesis that we can reject in some sense is I think, ill-conceived. These things are truly empirical. We do those studies to generate hypotheses and leads that we can then follow up.
But still, there is a question of what the level of significance should be.
Absolutely, and I think you should pay attention.
What's your recommendation?
Well, can't get away from these things. Well, I think there's a number of different ways of approaching the significance levels. There's certainly the more theoretically oriented ways, which Eric Lander and I have published on, and that Lander and Kruglia have published on, and other people have published on. You could use those as a quote-unquote guideline just to get you started. I think, though, if you go ahead and carry out a genome scan, and don't see anything that surpasses the Landers-Short and Landers-Cartwright criteria, it doesn't mean you throw away your study. Obviously, what you're going to do is take the highest peaks that emerge from your study and follow those up with more markers, or look in the regions where those peaks occurred for candidate genes for your study, or see if you can get corroborating evidence from another study done in another population for evidence for linkage, however vague or minor or insignificant in the regions that you found the highest peaks. So I think, again, these are more hypothesis generating, the genome scan approaches, but you can use the guidelines like the Landers-Cartwright and Landers-Short just to sort of put your results in a perspective, not the only perspective. Other ways of doing it, I think, that are non-theoretically based are via simulation methods. I think Joe had mentioned one, sort of the empirical approach. If you do a genome scan, not everywhere in your genome is there likely to be a gene influencing your trait. So whatever the behavior of the test statistic looks like in those regions, now again, you won't know a priori where they are, but if you do see test statistics bouncing around, so here's, you've done your genome scan and you've seen plots like these in all the genetics journals. You see the test statistic bounce around somewhat, and then it sort of peaks, and it bounces back down and goes there. Okay? Now obviously this area is giving us the most evidence for linkage, and whether or not it surpasses some criteria is, again, sort of a moot point at this point. What we know is there are other parts of the genome where a gene is not likely to reside, say right in here. What we see here is some bouncing around in the test statistic. These must be totally noise-induced fluctuations in the test statistic if, in fact, there's no gene here. So the behavior of the test statistics in these areas will probably give you some insights into how significant much larger peaks are. Now, of course, the problem is you don't know a priori if this is actually the location where disease gene doesn't exist and this one is. That's what you're trying to find. But somehow, using just the behavior of the test statistic that you actually observe to make sense of what's more likely to be a region of interest and what's not, it seems to be a logical thing to do. And people do this anyways. They do it by default. Sort of have, I think, a propensity to do this stuff. You look at what our data tells us and then try to draw inferences from that, rather than using very strict guidelines. So I think that's another approach.
I would also say, I mean, there are cases where using the guidelines are also unreasonably strict. I knew a genome scan that some people did recently. They used actually something like 600 markers, and they never found a log score over 1.5 in that whole thing. And I went back and I did looked at why that was the case and did some simulations, and I found that using the method they were using to do the analysis under the null hypothesis, you don't ever expect to get a log score over 1. So that 1.5 actually had like a genome-wide significance level that was something like 10 to the minus 4. I mean, it was almost impossible to get, solely because of the way the analysis was done. I mean, the theoretical results are based on a set of assumptions, and when you change them, the results change. For example, if you did the whole thing on cousin pairs with nobody typed in between, the variation is much larger, and those would be, you know, too low, if anything. And if you do a study in which, say, in this case, they did a parametric analysis with a penetrance ratio of about 10, which means that you're not using very much information per SIV pair. SIV pair may contribute 0.1 equivalent meiosis. In those cases, there's not enough variation to ever get to those high log scores. So that's why, I mean, I usually say in answer to the question that it's kind of like gambling at the track, really. I mean, you decide what your time and money is worth. And if you think a p-value of, 10 to 20% is good enough, then go ahead and do the extra work. I don't care. It's no skin off my back, right? If you want to go try to do molecular methods, it's your decision to make. That's the easiest way to pass the buck.
It's true that there is a bit of a catch-22 here, because I'm asked to do power calculations and whatnot for a lot of people. And the humorous anecdote that I always bring up is the times which I've approached about justifying a certain sample size. And so my obvious retort to the researchers as to do the power calculations is, well, what kind of effect size do you expect to see? And of course, the response is, if I knew that, I wouldn't be doing the study. You're definitely stuck in a catch-22. What you can do is say, for 300 SIV pairs and a marker every 10 centimeters, what's the likely effect you'd be able to detect if you make certain assumptions about the strength, disequilibrium, whatever. You can make those sorts of claims. And I think that's the way most people approach their grants. I have a certain amount of money or a certain budget or a certain idea about the number of SIP pairs. I need to justify that somehow.
That's the honest approach. The other approach is, what power would you like, not what effect size would you like.
Yeah, really. There is one issue that I'll just touch on, because Sue and I guess, are getting called out by the CAP, and that has to do with multiple phenotype information. Because I'm approached by a lot of people studying complex diseases like cardiovascular disease, where there appear to be kind of syndromes associated with the disease entity of interest. There might be obesity, diabetes, hypertension, high lipids, other sorts of things. Asthma in the same way. And the idea is, look, if I collect all this additional phenotype information, won't this increase the power I should have to identify disease genes? And the answer is not always. And so, for example, I could give you the following scenario. Let's say you're interested in, and this is a very important point, I don't think people appreciate this. Let's say you're interested in mapping genes that influence height. So that's your phenotype, you're interested in. And you say, I'm going to collect a heck of a lot of phenotype information. So what you do is you go out and you collect height in inches, height in centimeters, height in meters, in every conceivable way. And you think, look, I've got all this additional phenotype. But the fact is, it's entirely redundant. It's actually not giving you any further information about the activities of a gene. So then you say, well, that example is highly artificial. But if you go out and collect a lot of information, you won't know if it's redundant in that sense with respect to the disease genes you're trying to map. So there's no guarantee. In other words, if you went out and said, look, I've got insulin and glucose ratio, you're interested in diabetes, I've got other sorts of parameters that model some of the physiology behind glucose uptake and all sorts of other things, that information should contribute to higher log scores. And again, you may find yourself in a situation like the height in centimeters and height in inches. It's entirely redundant. You don't know that a priori. You're trying to use that information to make sense of the physiology and the genetics, but there's no guarantee that that information will contribute something unique and allow you to increase power with genes. You could find yourself in a situation like the height in inches, height in centimeters.
But I guess I would kind of say one thing to that. I think the only limitation is that I also wouldn't send out someone to only collect one phenotype if you don't really understand that phenotype either. The advantage of collecting multiple ways to measure that phenotype is if you don't have a good estimate of what is the best heritability, which one is the best measure used and has the most genetic component, or if you have the ability to look at it in a multivariate way, you could gain power. Right. So I wouldn't send everyone out either way. But the idea that you're just collecting one phenotype, if you have the ability to collect related phenotypes, not knowing which is the best to analyze, I think go out and collect it.
Well, again, it's this sort of empirical nature of science. I mean, if you go out and collect all these phenotypes, what answer might emerge from your results is that, in fact, they're all redundant. That would be incredibly useful. That'll shed light on what's going on. It could also turn out that those phenotypes are redundant, and then your log scores are 20 times higher than they would have been had you only collected one phenotype. So I'm not discouraging anyone from collecting more phenotype information. In fact, I'm a big advocate of it. I'm just saying that a lot of people have come to me, and probably Joe, and said, I've got all this information. Can I make use of it to increase the power of my mapping? Not always. You don't know if, in fact, you run into this height in inches, height in centimeters type situation where it's very intuitive that you want to increase the power.
There's also the idea that if they're unrelated, you might not map the gene for what you're interested in, but you might map something else. I mean, I always tell people when they're doing studies of, other phenotypes, well, why don't you get the BMI or something that you can measure for free? And well, if you find the gene, great, it's free.
And that's actually an approach that a lot of people are starting to take. If you go out and collect a bunch of SIP pairs, and you can collect other phenotype information, the genotypes aren't going to change. So you've got all that additional information to map genes. Okay, I think
yeah, I just want to take just a second before you go to thank you both very much for coming and just putting on a really good show here. I hope we can continue a little bit after we leave the session. Just show our appreciation.
Are there additional questions? We can keep going with it. Yeah.
Well,
I see a lot of the figures like are on the board, lot scores, locations. Using any kind of markers, the best that I've ever seen is that you come to a region finally where you've got something like 10 to 25 candidate genes in the locus. Well, yeah, or certainly a lot bigger, but that's the best.
Yeah.
Well, okay, then the question I had is, how do you go the extra mile and finally get to the gene? Without, do you have to totally sequence at that point, or is there something?
I don't even know what you do if you have the sequence. If you have a sequence of 25 centimorgans, I mean, there's going to be, if you have just a sample of 100 people, there's going to be, several things in there by chance that are perfect.
People.
Nobody's ever done it, because nobody's ever gotten to that point in complex disease. Nobody's ever mapped a complex disease with a genome scan and cloned the gene. It's never happened yet. It will, we hope.
Yeah. So it's only in those cases of rare alleles that lead directly to...
Rare alleles have easily worked. Candidate gene studies have worked, if you believe them.
Also candidate genes.
Sorry?
Also already identified candidate genes which are existing.
I mean, a lot of phenotypes there's candidate genes, like in HLA, it's the best example. I mean, it's the candidate gene, I guess, for everything, but it's been found to do something too. But I mean, I'm waiting for that answer, and I think everyone is. And my concern is that the answer is not technology. I mean, the answer's got to be somehow involved in, I think the answer is going to be more in what you sample, finding the right populations where, over a small region, even with a common gene, you might expect to have disequilibrium. And that's not necessarily going to be the case in expanded populations. The paper that I handed out to you guys is something we were doing to start to look at that. And the idea is even there, I mean, people have complained because you don't have defined haplotypes in really small populations, or rather you have assortments of associated alleles, but that can still only get you down to, say, maybe a couple of 2 or 3
Isn't that one of the rationales for the whole Human Genome Project, that then you could have...
There's a lot of rationales, but I'm not sure what the real...
Finally, your marker is, in fact, the gene.
But what do you do in this case? I mean, remember that, look at what these genes are like. I mean, you have millions of variants. I mean, if this is your gene,
I mean, no matter how close you are to the gene, you still have recombination until you're at the locus of mutation.
But even, yeah, but there's like this, I mean, this gene, these are all loci of mutation in that gene. And these all have effects on the phenotype. And this is where they affect it. In this case, there were 5 different classes, and there's, and this is a very simple gene. BRCA1 has how many 100 mutations characterized? And they're not all at the same site. This is what concerns me more than anything. And I think the models that I've been working on using these really small populations, it doesn't matter if there's 100 different mutations, you'll still generate disequilibrium as long as the population is small enough. But what do you do? I mean, I don't know. When you have 500 mutations in a gene, that doesn't work, because you never have the gene. You may have intragenic markers, but you don't have, the disease allele. In fact, you'd be very strange if you did have the disease allele, because if the disease allele is so common in the general population that it's one of the alleles that you chose to look at initially, what are the odds that it's actually going to be involved in the trait? Because presumably you're not picking normal, you don't pick the alleles to screen from looking at sick people. Another thing is that these SNPs and all this stuff they've been doing on that, as far as I know, have been concentrating on coding region things. And if you look at a lot of these Drosophila loci, a lot of the mutations are upstream. They're in regions that might be regulatory or some sites of binding or something, and who knows what exactly. I mean, we're targeting things without a good reason.
Basically trashing the whole Human Genome Project.
No, I'm not trashing it. I'm saying that the goals are overstated all the time.
Well, trashing in the sense that if there are so many variants of every single gene, and we're genotyping John, Harry, and Mary specifically, and they represent a subset of 0.1% of all of the different variants in the population, then what are we going to do with this tiny subset of genetic sequence?
What do you want to do with it? I mean, I think you can get to the genes. I just think that it's not going to be done
Well,
without.
I'm just wondering how the Human Genome Project got through scientific peer review.
Everybody wants the money. No, I mean, it isn't a bad thing to have these sequences, and we're going to map a lot of genes this way. Just, it's not necessarily going to be done as simply as everybody wants to make it out. I mean, we're not going to map every gene in 9 days, you know.
So what you're saying, technology?
Technology doesn't provide the answer. I think the answer is more study design, I think. This is just my opinion, that looking at the right study samples is the only way to do it, really. And looking and asking better questions. I mean, I think we now have the habit that if you see a disease and it occurs more than once in a couple of families you've observed, automatically, ooh, genetics, it's the thing to do, let's do it. Maybe, you know, in another 20 years, when people aren't so happy anymore, they'll be like, oh, genetics, we'll integrate it out again and do environment, and then they'll keep changing. And it's kind of, it's ill-conceived to try not to pick the right questions to ask and fund those and work on those instead of just doing it because it's a public health problem.
Okay, well here's another thing. We talked about doing the studies in animals and then going to look
Sure.
at humans. Okay, you take that approach and you say, here are 5 strains of mice that have this really low response, and here's 5 strains that have this particularly high response. And you take strain A versus strain B over here, and you get these, you find these regions of candidate loci. If you take strain 2 versus strain C, you find 2 entirely different regions of loci with candidate genes. And you can take any 2 pairs and you find entirely different regions.
Yeah.
Well, does that mean in every single, well,
Yeah.
I mean,
How many genes are there?
then you lose your confidence that when you're looking at humans, that you have any chance, if you do find a candidate region, you figure that's maybe 1 candidate region out of 1,000. Then
Sure.
the 1,000 that you don't see
Could be.
may be the most important.
My favorite question is one that Nick actually said he's working on now, and that is, I want to see someone take these genes that they map in the mouse and the rat and try to map them in the mouse and the rat if we're using SIB pairs. Go out and take outbred mice and see if it works there. And if it doesn't work there, where you can control environment, you can control everything else, you might want to think twice before immediately going to look at the same corresponding genes in humans. But nobody wants to do that, and he said that basically the response he got from people when he proposed it was very similar to what I got, which is, well, if you want to map genes in mice, why don't you do a backcross? The point is not that. The point is to see if the approach really will work and what kind of sample sizes you really need with real data, as opposed to using some model that we come up with because it's mathematically tractable. Well, those are the models we're going to map them for, and they, you know, it obviously tells me our models are wrong. The fact that we haven't had so much success.
No, but what I'm saying though is if in inbred mice, well, depending on which 2 strains you choose,
of course,
you get entirely different loci. Then if you go outbred, then it's an impossible situation.
But we are outbred.
That's the point.
Unless you pick the right population that isn't. I mean, there are populations
Right,
in the
and
world
so
that are
you
better.
choose your little island population, but that
They
could
don't have
have no
every phenotype.
relationship to anybody else in the world, just as these 2 mouse strains could have no relationship to any other 2 mouse strains.
Of course, nobody ever says that they will. I mean, I think sort of the idea has been also they look at these rare early onset forms of some of these diseases, like I guess there's early onset glaucoma that's real severe that they found a gene for, or this and that. The idea I always understood was that they get some idea of the physiology and the system that they're working in, and they can maybe understand what's going on a more biological level. But I don't really think people expect that they're going to map all 100 genes involved in each trait. They're hoping to map one of them. I mean, if we found, you know, 1 gene that we didn't know about that was involved in diabetes, we'd be happy. But so far, I'm not convinced that they have, except, you know, obviously insulin and HLA, which are obvious candidate genes.
But if you find 1 gene, like in the hereditary non-polyposis cancer, if you find 1 gene, then you can look for others in that same pathway.
Sure, sure.
I mean, it leads to
right, exactly, yeah.
To a whole
hopefully
set of genes.
Hopefully.
So, because it's the pathways that are important after all the biological.
We hope.
Yeah. And I don't understand why you're bothered about there's so many mutations like in BRCA1. I mean, all of those mutations, just like in TP proteins, probably this example, like
Okay.
With any mutation, I mean, they all are affected with protein.
Sure.
In a critical point.
Sure.
Activated.
Sure.
So, I mean.
But they affect your ability to find linkage
Right.
disequilibrium.
Right.
Not
So-
linkage, but linkage disequilibrium. That's the key, is you
Right.
can
Yeah.
get close, but you can't find it.
You can get
In
linkage.
retrospect, you're
Right.
saying you know p53, but we're saying if you get linkage and you get close, okay, you can get to the point where you're not getting any, you're not picking up any recombination, but you still don't have the gene. You need something else to drive you closer to actually find the gene.
Yeah.
Right, but
Okay.
that's where the technology will help, right? I mean, it's easier to do
Well,
things like.
technology
But
will
I
help.
guess that's the question, how is technology
Yeah.
going to help you? You see, I mean, you're not, but the idea was to use technology to pick up linkage disequilibrium, but you're not
Okay.
going to find linkage disequilibrium if there are thousands of different so-called ancestral mutations that have all
Yeah.
been occurring, and everyone has a different haplotype. Linkage disequilibrium is not going to guide you in, and you're going to miss it.
I reckon I was thinking like with the BRCA1, I mean, but if you're working with families,
But
each
what-
family would have its
Yeah,
own.
right,
mutation,
right. But in each family, the
What
regions
led you
could be
to
just large.
BRCA1? But don't forget, what led you to
Yeah.
BRCA1 in the first place? Mendelian families.
Yeah.
Okay, it wasn't that you found it by collecting generic breast cancer families. It
Right.
was Mendelian families that looked autosomal dominant. Recombination drove you.
Mhm.
I mean, but we're not, for most of the complex genome screens in these common diseases, we're not using recombination to
Mhm.
drive us toward a gene. We're
Right.
using this idea of identity by descent, which doesn't have as a parameter of the model or model free, however you want to think of it, doesn't have recombination as part of the parameterization. So you don't have it to drive you toward a gene.
And in fact, your peak.
You get these big broad
Yeah.
regions. I mean, you get broad regions because you have markers with variable heterozygosity, so you're not getting the same amount of information.
And because
But
also the amount of sharing you expect at the true gene is only maybe 60%. And if it's 60%, there's a, basically a 40% chance that you have a higher peak on this side and another 40% on this side. And I've seen in simulations, you may have the maximum be 20 centimorgans away.
Which is why people are not finding. I mean,
Yeah.
it's not led to the discovery of a gene.
Yeah.
I mean, I guess I'm not quite as negative as Joe is because people have not been doing this for that long either. I mean, the technology has come. I mean, I'm somewhat negative. I mean, I'm with you on some of it, but I think you can't say the whole, it's all hopeless.
I didn't say it was all
I know you
hopeless.
didn't, but I think you led many people
I
in the
mean,
room thinking.
let me ask y'all, here's the other question, I mean, thing that bothers me sometimes, all right, so, like even with, breast cancer or colon cancer, I know the cancer's better, you have heterogeneity, which is different than a complex disease, the way I understand it. Heterogeneity just means one family will have one gene, and another family might have another gene, but in each family.
Complex only means we don't know.
Yeah.
Right, all right, so how do we know that things like diabetes, that if you really, I mean, that you don't have this.
If they get the large
heterozygous,
families, then we'll be able to
instead
tell.
of really, it calls you having many genes in the same family, it's just each family might have these different genes, and you're confusing it, that it's multiple genes where it's really more this heterogeneity.
But there's no difference in terms of how we get there unless
Yeah,
you
but
get the right
biologic,
samples.
but I mean, but you still know that in maybe in a given family, it's only one gene. Okay?
But that's the
It's
problem.
not multiple.
But how do you
You
find
don't
that
know
out?
Well,
That's
that's
the
what I'm
problem.
asking
That's the
y'all.
Oh, yeah.
You have to distinguish
It's so
between-
common that it's not without the realm of possibility, that you get two founders at the top of your pedigree, each of whom has diabetes for totally different causes. I mean, it's the idea of genetic heterogeneity. You get two deaf people who have deafness for two different reasons. Okay? And now try to segregate and find the genes in those families, and you're not looking at informative meiosis.
And if a disease is 10% frequent, you have a big pedigree with 30 founders, you probably have 3 people that are founders who
Some
had
people have
it.
tried to go to early onset diabetes. I mean, I'm part of a project where they're trying to do Pima Indians, but it's so common that you have so many genes flying around in there that you can't straighten the thing out. I mean, people have tried these different angles of trying to get some kind of an isolate. The idea is to try to get as much genetic homogeneity as you can. Okay, can you make a homogeneous group? Whether it's by limiting the number of genes in that sample, and people have tried to do that by going for Mendelian families, because they think they're getting something that's simpler and cleaner. But the experience has been that these Mendelian families, you get a single gene major effect, it's not having a big effect on garden variety Alzheimer's. It hasn't proven to be all that effective. It's helped them find other genes that have actually gene similarities that are also causing early onset Alzheimer's. It's very wonderful for that, but hasn't given us a lot of insight on Alzheimer's. And everyone, I think, has lost some perspective on ApoE being, a risk factor for late onset Alzheimer's, but it was not found, you know, through a complex genome screening. I mean, it was fortuitous.
Yeah.
And a lot of people try to use that as an example of a complex gene success, and it really is a fortuitous discovery. And other than that, that's it. I mean, do you agree, Joe? I mean,
no,
I mean, you have to put it in some perspective.
Yeah.
The tools we have, I mean, are not there yet. I mean, most people have drawn the conclusion that our samples aren't big enough. You know, we want to get more homogeneous, but we haven't figured out how to get there.
And I kind of worry, though, about the idea of making the samples larger and tooling. I mean, unless there's a reason. I mean, I know a lot of people are doing it. I just worry, what happens if 10,000 SIP pairs isn't enough? Yeah?
Because if you haven't made them any more homogeneous,
you're making them more
you haven't
heterogeneous.
You haven't helped it. Unless you go through some strategy to make them more homogeneous, whether you go for earlier onset, you go for bigger families that have more affected, where you think it's more genetic. You go for families that are unilineal. You know, you won't take it unless it's only on one side of the family. I mean, you got to have some angle to make them more powerful. And in essence, you're trying to parameterize them.
But this might be getting another phenotype. Well, this, you know, I mean.
Well, I kind of think that the best way to do it is pick a population that you think is good to study, and then instead of picking one phenotype to study, just study a whole bunch of phenotypes in the same sample where the sample's huge.
Related phenotype.
Not even related. I don't know why they'd have to be related. I would say, I mean, if you, for example, took Greenland and genotyped everybody and phenotyped everybody, there's 50,000 people, but you phenotype everybody for whatever you might be interested in, then see what washes out. I mean, because I mean, at least there you have more, you throw in more darts at the wall, and one of them is going to hit your target, hopefully.
Let's pick a little island in the Pacific that people you go to.
Well, but also more people have gone there, you see, because it's right.
That's right.
Would you like to comment just on a different note about what future do you see of the Bayesian statistics in all this?
I mean, honestly, no matter what approaches you use, it doesn't really make much difference. I mean, we're all sort of assuming something a priori, right? The Bayesian just means they're assuming some more stuff, or they're trying to assume distributions of stuff. I mean, I don't really think that the statistics matter. The statistics we use have not changed since the Lod scores in '55. I mean, we're not doing anything today statistically that we couldn't have done 50 years ago. I mean, the solution isn't coming from us, if you mean, is there a better way to analyze the data? I mean, we've all been working on it, and you know.
You're being modest.
No, the system is so simple. Genetics is simple. It's Mendel's laws. That's it. There's not much beyond that. There's genetic laws, and there's like genotype-phenotype relationships, possibly including stuff about the environment. But they're simple. I mean, there's not much we can do about that. You can just try to get the right sample to study. I mean, really, it doesn't matter. I mean.
Well, tell us how to get more R01s then.
Well, I mean, I think part of what people are also suggesting is you might want to go to some kind of a multi-strategy approach. I mean, collecting SIP pairs is not, I mean, I think what Joe's trying to suggest is you're going to need different kinds of samples with different kinds of features potentially, and maybe that will help you get closer. I mean, I don't think anyone has.
I mean, my biggest concern is like, I mean, sometimes you see papers like, I mean, this one where Neil's just saying that you want to collect 10,000 SIP pairs and do TDT on them. But by the time you get 10,000 SIP pairs, if the prevalence is 10%, the population you're sampling from is, you know, it's already, what, 100,000, right? And that's, I mean, as you go up, and you want SIP pairs even, I mean, the probability of getting 2 SIP pairs is going to be even smaller, yeah? If you get 100 SIP pairs, the probability if it's 10% prevalent, you know, it might be, you know, if there's a relative risk of 5, you still would be up around, you know, several 100,000 minimum. And I doubt that anybody's going to a homogeneous population of that size and getting them. They're just getting them saying, okay, and this is true. I mean, I know people who are doing this, and they say, well, everybody says we just need numbers, so we can't have numbers and the right population, so we're going to go for numbers. But when you go for numbers, I mean, what's the point? I mean, if you're adding more, you're adding as much noise as you are signal. Yeah?
Everyone appreciated that point until a lot of, I mean, the last couple of years of work has led to the collection of samples that have sort of proven this. I mean, I'm not sure everyone realized this. I mean, would you agree? I mean, I don't think everyone realized this 5 years ago. I mean, some of this has been trial and error discovery.
Yeah, I mean, 5 years ago, the idea of getting
this
300 SIP pairs, we thought, well, no one's going to do that. You know, no one's going to spend that kind of time in doing that. Now when people start talking about thousands of SIV pairs, you really have to say, wait a minute, that's going to be really expensive. And is it going to give you something? Under what conditions does that actually solve the problem? I don't know. Because as you said, yeah, the more large sample you get, the less identical they are.
So don't you think all of that, a problem arises out of people just not being able to talk to each other? So that if the statistician doesn't know any genetics and he's just doing the calculation, or if the geneticist
I just think we all would, it's very nice. I mean, I haven't been in the field, neither one of us has been in the field, more than, 5 to 10 years. I mean, I don't think most of us realize how complex it is going to be. The number of genes you'll be talking about and what looks like number of genes of relatively potentially small effect. We all thought, we all hoped that there would be a couple genes, big effect, and then a whole bunch of little ones. You find the big ones, you find the little ones. And I don't think
well, this would make jobs longer.
Yeah,
we don't have to retire
it doesn't look as promising as that.
So you think it's just a matter of...
Well, I think it's been trial and error. I mean, we all made assumptions on our initial power calculations, and I don't think most of them, some of them were right.
Yeah, I think the biggest problem, I think, because there's so many people in genetics that don't necessarily, I think either they don't understand all of the stuff about the statistics of the molecular biology, or either other side, as you suggested, or they're not necessarily very interested in the process. They're interested in getting the genes, and they don't really care so much about the process. I mean, it's kind of gotten out of hand.
Well, and part of it probably also has been just to add, the whole thing is phenotyping. I'm not sure many people necessarily always realize how important phenotyping would be. That, I mean, your genotype is only as good as your phenotype is, and that's another way to get to a more homogeneous population. I mean, I think we went into it relatively naive that, if it looks like it, it must be it, and it's okay to throw them all together.
And the one thing I think is a real big problem, I mean, genetics especially because of this interdisciplinary nature, perhaps, it's a bandwagon thing. Everybody jumps on whatever the hot bandwagon of the month is. I mean, either it's, okay, now every paper must have, this Gene Hunter NPL score, or now every paper must have, your favorite linkage disequilibrium statistic, or now, the only way to do this is find an isolate that's gone through a big expansion and we go and look for haplotypes, or maybe you don't want the isolate, you want some other thing, but everybody tries to do the same thing, because somebody, whoever it is in each case, gets out and says it, and they say it often and loud, and everybody thinks, okay, this guy said it, therefore it's true, I'm going to do it. And it's led to more, wasted time and money, I think, And now it really worries me when, Francis Collins goes on TV and starts talking about how they're going to be able to do everything in the next 10 years, our medical thing's going to change, and we're going to have all these genes understood, and we're all going to live to be 500 years old, and it's crazy.
Simplification.
Yeah. And it's scary because that's what the public is starting to think, and that's probably why they're as concerned as they are about some
Although it did lead to larger NIH funding, apparently.
Yeah, but will that continue if we don't live up to their expectations?
Yeah, that's exactly the same thing as the war on cancer.
Yeah.
That you guys are too young to remember. But there was this unrealistic expectation. It drove the funding, and actually it still drives the funding. It didn't deliver.
I want to have a job. I don't want to be unemployed because people promised things that couldn't be done. Yeah.
Yeah. Just it might be very naive. When we do SIV pairs, mostly we talk about affected SIV pairs. What are your thoughts about completely discordant SIV pairs in this kind of process?
Depends on the model. Everything depends on the model.
Well.
And if you find them, you got to be sure that they're discordant for the right reason.
And that they're full siblings.
Yeah.
Are they so discordant because they're not full siblings?
Yeah, we actually had a study that someone came to us. They gave us the data for these families. It was for some very common trait, and this was done by some guy who had a clinic, and he had all these patients, and he gave them to us. And it was like 100 SIV pairs maybe, with the parents and everything. So we genotyped them for a couple candidate genes trying to replicate something, only to find out that there were inconsistencies all over the place. And we went back to the guy, and he said, oh yeah, well, yeah, these are adopted, and these are house SIVs. He knew it.
Common environment.
He knew it, and he gave us the samples to genotype with the money. Another one that I really like is another person who I don't want to mention,
Yeah,
came to us.
Videotape.
Yeah, right. Another person came to us who I don't want to mention, and she had just sent off some samples to research genetics or something to have them genotyped, and she had it was a rare recessive, very inbred, nice, neat pedigree. She happened to have one SIP with 10 affected, so she sent those 10 and the parents off to be genotyped. Came to me complaining, obviously my programs were wrong because she was getting lot scores 0 everywhere. The parents were both affected, and it was recessive. So, I mean, it's a very real problem that this is what's going to judge our funding in 20 years.
Other questions?
Well, since we have a little more time, I should keep asking. Well, this question, maybe I alluded to it, about having controls. By adding another set of new controls, and then we pick, does that in any way, in your mind, increases power? Like 1:2 match or 1:4 match, as we have in the epidemiology.
I mean, I guess it certainly isn't going to decrease your power.
That's a lot of work.
I mean, you have more accurate estimates of the allele frequencies, I mean, obviously more samples are better, but I'd rather have more cases.
More effective, yeah.
Because remember, the controls here are only being used so you get the null hypothesis distribution. The real question is, are these affecteds more similar to each other than you would expect? So the controls are just, what do we expect? I mean, I reviewed a paper once where they were looking at something, and they were saying that you could take all the uninformative meioses and assume they were informative with the same probability as the other meioses. And I said, well, in response, there's 5 billion people in the world that don't have that disease, so you can multiply your lot score by 5 billion.
I guess I would just give our plug that the next workshop we're talking about doing will be in April, and we're going to look at QTL mapping in model organisms. So we'll be moving it to the animal realm, but I think bringing up actually many similar questions and trying to see if animal models give us any